Atamaria ki a tātou katoa I tēnei ata, huri noa tēnei tō tātou whare, tēnā koutou, tēnā tātou katoa. So let us begin our conference with krakia. Kia whakatauha, whakatauha. Whakatauha te rangi e tūnei whakatauha, te papa e tako tōne. Titia ke oho, titia ke awātea. Titia ki te poho tāne nui ārangi, whano haramai te toki haumi e hui e tā ki e. Ka tira kia tuku whakamoimiti ki aia ki te wahingaro. Kia ino I tātou. Ei hoanga mano ki te rangi te tihi o tau ātea ki te mata o te whenua tau turanga wāwai, kei ngā wahi katoa rātau Claudia Tanganui. E te matua e tamata wairua tapu me ngā koutou ana hera pono. Kia tatu mai ana rā koutou ki wainganui tēnei hakamoimiti o mātou hei tātari I ngā kupu katoa, hakapoa ki nei a mātou I roto nei tā koutou Claudia Tanga. Ko koutou nei ko te arepo te omeka, tīma tangata whakaotinga mai o a koutou mahi katoa e tūmana ko tia nei I roto nei tā koutou Claudia Tanga o tira ihua. Tēnei rā mātou auponanga I tia tūkolopiko nei mātou ki mōtō aroro. Tuku ake nei ko te reo whakamoimiti Claudia hono re me ngā whakawhetai. A ka whetai tu nei mātou e iho kia koe, mō umanākitanga kia whakataungia ai a koe ki runga tēnei tā mātou hui I tēnei ata. Me ngā rā e tū mai nei ki mōi a mātou tēnei katuku e nei inoi ki mōi a koe iho. Tu a uriuri whai o ki katona te rangi te whenua ki tau Claudia Tanga, ko I rā tēnei ka whakahoki atu rā ngā mihi tuatahi kia koe. Nā reira, e te matua I nā nei kia koe I runga tonu tau ingo tapu tau ingo kaha. Whakaungia mai mātu I roto tu koutou aroha mutunga kore, pai here tia ki te rangi māri e I ngā wā katoa. Kei roto tonu ko koe te māngai te kai āwhina tautoko mai, ai ni aka nei. Ai. A tira hea tātou katoa I tēnei ata. Nau mai, haramai. Haramai rā koutou ki tēnei tō tātou hui huinga. I tēnei ata, mō ngā rā e toru kei mo I a tātou. Nō reira tēnei te mihi atu ki a koutou, koutou katoa ko tai mai I tēnei ata. Ladies and gentlemen, good morning. Good morning and welcome to this Bioresearch Centre conference here this morning. And I am sure you'll find it of interest, some certain things that certainly will enhance and support what we do here within this particular call. I'd like to also say at this present time, this is most probably and likely to be the last conference under this tag of Bioprotection Research Centre. So it's a very special event here for us and for you, no doubt. Nā reira, acknowledging you all for giving up your time to be here. Nau mai, haramai, welcome, thrice welcome, tēnā koutou, kia ora tātou katoa. Haramai, Jason. Michelle. E te hau, horoia, horoia, e te ua. Whiti, whitia, haere hau hake au a kōrero kia rātou. Koutou te hunga ko hori tōra mai rā koutou kia mātou. Koutou haere, haere, haere. So we acknowledge those dear to us, and in particular some members of the staff of this university who are no longer with us. We acknowledge them and we farewell them, though they are not entirely lost to us, but continue within our memories and their contribution to this university, those who have gone on. Ro reira tāpiti hono tātai hono koutou te hunga ko oki oki koutou kia koutou haere ngā mate haere. Āpiti hono tātai hono ko tātou rā tēnei. Ngā hui huinga o rātou mā huri huri no tō tātou whare tēnā koutou. Ngā maunga katoa kei Wainganui, acknowledging you all from whence you may come from, your mountains, your awa, your rivers, and in particular, your presence here over the next 2 days. Tēnā koutou, tēnā koutou, tēnā tātou katoa kaheke.
Hi everyone. I would like to introduce the current chair of the Bioprotection Research Centre, John Rodwell.
Kia ora, good morning. My name is John Rodwell and current chair of the soon to not exist Bioprotection Research Centre. To be superseded, and we'll talk more over the next 2 days about its successor, Bioprotection Aotearoa. But in my capacity of chair of this organisation, let me say it's a great pleasure to be here. I'm inspired and in awe of some of the faces and intellects that I see around the table. And as I said to my colleague Don Hammond as we came in, at our stage and age, isn't it wonderful to put 2 days aside and indulge ourselves in learning? So I hope you all share that same exciting aspiration for the next 2 days. But let me just give you a little bit of context to set the scene today. My life is really orientated around agriculture and the food and fibre sector. I'm a farmer and a businessman, and in a wider context, I have the great pleasure and opportunity to be a director of Manaaki Whenua, and now one of the challenges, the Bioheritage Challenge. The food and fibre sector in New Zealand is profoundly important for Aotearoa. It occupies 14 million productive hectares of land. It accounts for 75% of the country's merchandised exports. It represents over 19% of GDP, and it accounts for 1 in 10 jobs in the country. Our country's reliance on agriculture and the food and fibre sector is unusual for a developed country. This reliance means that in order to maintain living standards and grow our economy for the future, our sector will need to grow by at least 2% per annum for the next 40 years. Now, as we know and increasingly are confronted, this won't happen from production increases. Our productive landscapes are fully utilised and in some instances over-allocated and threatened. This will only happen from increase in value from our existing agricultural landscapes. So the value that we seek to enhance will only happen by firstly ensuring we maintain the health and productivity of our productive landscapes, keeping them free, protected from weed, pest and disease incursions, and secondly, by doing this with integrity and in ways that increasingly resonate with consumers. For example, reducing the amounts of synthetic pesticides and fungicides and herbicides used, and in some cases replacing them entirely with systems changes or with bio-alternatives which are far more socially and consumer acceptable. BPRC was established to specifically develop the science and education pathways that will help us understand, protect, maintain and enhance the health of our productive landscapes and our Indigenous landscapes, and to create pathways for young scientists to continue that work well into the future. Some of the examples which you will be familiar with of what we consider to be our successes over the last few years relate to the work we've done in fundamental science development around disease, pest and disease incursions. Notable is the work that's been done in and around Kauri dieback, Argentine stem weevil and of course PSA in the kiwi fruit area. This is part of a raft of fundamental science that's underpinned our purpose. In addition to that, this organisation in its lifetime has produced in the order of 150 PhD and Masters graduates, all of whom have gone into the future to continue this profoundly important work. And of course, associated with that, as organisations like this do, the publication of enormous number of science journals and papers which have informed both our industry in New Zealand and industries around the world. So it's my pleasure to welcome you here today. Our purpose has been, as I've outlined, very much to understand, protect and enhance our productive and Indigenous landscapes. Our purpose going forward will be to do that with further vigour and importantly to encapsulate the profound value and relevance of a Mātarangi Māori framework which will inform our direction in the future. So to all of you, thank you again. Thank you for being here, and I hope we all will learn, and I hope we will all learn from each other. I appreciate your time. Thank you very much indeed.
Tēnā koutou katoa, ko Travis Blair aho. I'm the current director of the Bioprotection Research Centre for another 7 days. And then Amanda Black and I co-direct Bioprotection Aotearoa going forward. Now, I had very carefully left a lot of time to make sure that we didn't overrun this part of it, and we've got 15 minutes until the first talk is supposed to start. And I have no idea what to do about that. So I'm going to take a long time doing the housekeeping. One of the first things to mention for housekeeping, there is an issue in Wellington at the moment with COVID, with an Australian visitor who had COVID in Wellington and wasn't detected when we went back. We have made the call this morning to continue with the conference, hoping that's the right call. There are some people from Wellington who are coming. I don't think they're here yet. There will be an announcement at... Yes. I'm sorry, it's one of those areas that we're really caught a bit short this morning about what to do about it. But there will be an announcement around 1 o'clock as to whether there's any lockdown in Wellington, and we will act accordingly on that. So I would ask anyone from Wellington, sit in a corner. So it's about all I can think of, I'm sorry. So anyone who's uncomfortable with that, please, look after yourself and your health, and leave if you need to. So for other housekeeping, with that in mind, QR codes, please scan them when you come in. The emergency exits are marked down the front. I don't think that is one. So two at the back and one at the front here. So, and Sandy, who has been organising this with the green hair, she's the warden, so follow Sandy where she goes. So, morning and afternoon teas will be in the foyer out here, and lunch will be in Mrs. O's. Breakfast for people who are staying in campus accommodations is in the dining hall in Mrs. O's behind this building, and that's from 7 a.m. Tomorrow and 8 a.m. on Friday. At 12 o'clock at the finish of this first session, we'd like to have a photo of everyone If it's not raining, we'll assemble in front of the library building. If it is raining, we will go to the next lecture theatre inside this complex. For the conference dinner, we're going to Larkham's Vineyard, which is about 10 kilometres from here. Buses for Larkham's Vineyard will leave at 5:15 from the Springs Road car park, which is that way, opposite Gate 3. So it's the overflow car park for those at Lincoln, on the same side as the accommodation. They will return to the same place at the end of the dinner. Security have been advised that there may be cars parked in those Orchards and Burns car parks for a longer time on Wednesday evening, so you don't have to worry about leaving your car there. Any problems? See Sandy. And I have a phone number for her, but as none of you are going to write it down, I won't bother. And if you are tweeting or posting on social media, please use the hashtag Bioprotection21. So, that leaves us with 15 minutes. I will say, because we're live streaming and we want to keep to that time schedule, I would say, please talk amongst yourself for 15 minutes and I will be back. Gail.
Sandy was talking about organising a bus into town after the
Sandy, would you like to come and say where that's got to?
If there is enough call for it, yes. I'm not going to do it for 4 people though. I want to show of hands. Yeah.
Oh, sorry. No, this is a separate issue.
Message.
So, does anyone want a bus into town after the dinner? 1, 2, 3, 4, 5. Sandy, there's 6 or 7 at the moment, and that's not everyone who's coming. Yes, there will be. Any other questions before we have a slight recess? Thanks a lot.
Trace.
Hi everyone. Just one more piece of housekeeping before we start. For those who are looking to get onto the internet, apparently it's Open Campus. That is the network you want, and you put your cell phone number in and it sends you a password. At least I've just been told that's how it works. So please let me know if that doesn't work, and we'll find you another way at lunchtime. Well, now I've got your attention, I may as well start. So this conference is really a mix. It is a celebration of the last 5 years of our Bioprotection Research Centre program and a launching of Bioprotection Aotearoa. And you'll notice in the program that some people have BPRC and some people have BA after their talks, indicating which of the two Centres of Research Excellence they are representing, and of course some are representing both. But with that in mind, I wanted to start with the first talk of actually just looking back on what we've done in the last 5 years as a core, and a little bit of a reflection on what has been 18 years of being the Bioprotection Research Centre. So, oh, and don't forget the hashtag. And Margaret, lovely photo of gumboots, thank you. So just a quick recap on why our area is so important. And I think John put it very eloquently that basically New Zealand is an agricultural exporter. We trade on both our natural environmental beauty and our produce. And so the top line there for me is the most important. And it is the question of why have a core in this area? Why have a Centre of Research Excellence in plant protection? And it's because New Zealand is unique. So even though we have exotic plants here, our combination of our soils and our environment, the plants we grow, the pests we have, are unique. So no one else is going to do it for us. So with that in mind, the National Centre of Research and Excellence, which now is the Bioprotection Research Centre and will be Bioprotection Aotearoa going forward, was established in 2002, first started in 2003. It's a centre of research, and especially fundamental research and postgraduate training. So we are set up under the Education Act, and we are funded as an education facility. So we're not funded out of MB, we're funded out of the Tertiary Education Commission. Our current mission for this last 5 years has been the sustainable reduction and prevention of damage to our land-based plant ecosystems from invertebrate pests, weeds, and plant diseases. So very simple message. For some reason it's taking me 2 to do. One of the things that we're actually quite proud of is that over our life, we have continually added partners, and we started with 4 partner organisations in 2003. Scion joined us in around 2013. In this last few years, both Otago and the University of Canterbury have come on, and going forward, we're adding 4 more institutes in New Zealand. And so we're starting to really cover the majority of the research institutes in New Zealand working in our space. Now this is very significant given we didn't get any more money. And so people have been willing to come and join us, not necessarily for more money, but to be part of the family and to be part of the effort. And the efforts of cause go beyond that small amount of money that we do get. And it's the joining up. It's more than the sum of the parts. A little bit about how we actually developed the program that we've got now. And it's on the back of, in 2013, this is what we looked like. We were doing a lot of work in a lot of projects, but they become quite fragmented. They were all really good pieces of work, but you were not seeing that synergy between the projects that really a core should be. And so we had a big think about this, and we thought about how would you actually shape a program that made more sense in terms of where you wanted to be. So the first question is, where do you want to be? And so we looked at it and we said, what are the big questions that we could work on? First one, what makes an organism a disease? And it was around the time of PSA. Why was that strain of Pseudomonas so effective compared to all the other conspecifics? how do organisms evolve? And we looked at this as real-time evolution, and I'll give you an example of this later. But basically, it's happening all the time. How is that driving plant protection? Then, why are some species invasive? Why is New Zealand suffering from some weeds, for example, and not others? Why are some of our insect pests big pests and not their conspecifics? And then finally, and this is an important area for us, is how do all these organisms interact? So rather than just thinking you're going to go kill a pest, what happens in that entire environment and how do you make that better? So we broke that up into 7 projects, and these were large multi-institute projects, all set up to run for the full 5 years of our funding. And this is what I'll be reporting back on today at a very high level, just some examples of where these projects got to. So, contemporary weed invasion, loss of effectiveness of a biocontrol agent, plant virulence and plant defence, enhancing beneficial endophytes. Why am I reading this? Because you're all quite capable of doing that. You could put it around several areas. So rapid evolution. So we had 2 projects which were really mainly about evolution. What makes a disease? We had 3 projects that were in some ways touching on this, why are some organisms disease causing and others not. We also had an overlay of making better biocontrols, but this is fundamental research and how could that lead to better biocontrol? And then the whole bit at the end of what does the whole system look like? So what I want to do, and coming this way, what you see is that our model for operation is fundamental research using these big teams, mainly PhD and postdoc driven. They're the engine room of what we do. But then those results get taken up by our partner institutes in more applied programs, and that leads to our impacts. And of course, for any fundamental research, you're looking at impacts somewhere between 10 and 30 years out. It's very strange. Oh, there we go. Right. So, I'm going to step through project by project, just at a very high level of what was found. Phil Hume led this project on contemporary evolution in weed invasion. And what this was looking at is, of the major theories around why weeds become invasive in New Zealand, what was actually the case. And using Rumex as dock weeds as the model system and looking at 4 species of dock, lots of different experiments, including some GBS studies and looking at the plants in the UK, for example, and in New Zealand, and trying to work out what was going on. A couple of take-home messages. Rumex, in one of the major species, its enemy escape. They actually didn't bring the pathogens and the pests with them, and that has led to them being far more successful. It's not the case in all of them, and I'll come back to that in a later project. It wasn't the result of increased genetic admix, it wasn't the result of hybridization. And so it's not that these weeds were mixing and matching and becoming more effective, but rather the stronger environmental selection pressure in New Zealand that these dock were able to respond to and were selected for better effects. And so there's a bit of increased phenotypic plasticity there. So, of course, 5 years of research. Anything I say is going to be really underdone, but if you're interested, talk to Phil. And the same with all the project leaders. But it isn't just about the research that we put out. What's the so what question for all of these is important. So dock weeds can adapt rapidly. They're drought resistant, so they're going to become a greater problem under climate change. They suffer less damage from herbivores and pathogens, so we're not going to see any sort of natural control going on at a level that's going to make a difference, which explains why they're a weed now. So one of the take-homes from the project is this is a perfect example where some classical biocontrol or inundative biocontrol, where you put something on massively as a biocontrol agent, is the way to go. The second project run by Steve Goldson, led by Steve Goldson, was a really interesting one in that it's one of the few examples in the world where an introduced biocontrol agent has lost effectiveness. So this is a parasitoid wasp brought in the 1990s from South America to control Argentine stem weevil. Did a wonderful job for 15 years or more. But when the group went back and looked at the data, which is the graph here, that's 23 years on the graph, the parasitoid has become overall less effective. And so the team set out to find out why and what could be done about it. The weevil is sexual, the parasitoid is asexual. The weevil has evolved a mechanism of avoidance. That means it's not being parasitized as much, and it's quite significantly not as much in some cases. And so it's patchy. Not all the populations around New Zealand have been under the same selection pressure, so you're not seeing the same outcomes. There was also what Steve used to call the grand experiment when he first released these parasitoids back in the 1990s, released 7 ecotypes collected in different parts of South America and put them out there. They've now been doing a lot of genome sequencing of these parasitoids, and they can start to tell these ecotypes apart, which could never be done before. They were always looked exactly the same in any of the molecular techniques. So the so what on this one is basically, can we find which of these parasitoid populations has actually survived and thrived? And so is there an ecotype that should be released in these other areas? It also helps to define what traits will make a better parasitoid going forward. And then there's some very interesting concepts around, and Peter Dearden's been leading some of this thought, around whether you can actually switch these asexuals back to sexual in containment to breed new lines and then switch them back to asexual and release them. And so it goes all the way through from understanding biocontrol to even coming up with better approaches. Project 3, led by Matt Templeton, pathogen virulence and plant defence. And this was really about using PSA, Pseudomonas, on kiwi fruit and Phytophthora on kauri, and looking at some of the fundamentals of disease. So why these particular strains were so effective. Looked at the epidemic origins of both. Some of it is a little contentious because two groups can come up with different evolutionary patterns for these. So when Phytophthora actually emerged in New Zealand, for example, we're still unsure. There's 2 ways to do it. They come up with different answers. But that's good. That's science. That's what we're trying to do, trying to understand why you might get a different answer. Some of the other work around the evolution of PSA was more straightforward. One of the big findings there is that the mobile genetic elements, the things that jump around inside bacteria, are driving the evolution. And so it's a relatively random event that will generate these strains, but it will happen. A lot of work on things like effectors. So the bits that are how PSA and the plant communicate and how that's driving the virulence of these organisms. And that leads to some ideas about what you might do about it. And there was also some work on Phytophthora and kauri tissue and how genes express there. Obviously, this is all very high level I'm giving you, and I'm feeling a little bit too high level, but... So, where do you get from that, the so what? Better understanding of how these organisms are evolving in real time. Bacteria evolve fast by comparison to plants, and so you've got to keep that in mind. These genes are jumping. How is that driving? Where are these next diseases going to come from? What should we be worried about? And a couple of just practical things that came out of this were detection tests. So developing LAMP assays for Phytophthora or PCR-based assays for the Pseudomonas. This really improves field prediction and field modelling. And so even at that level, what comes out of fundamental research can really help the applied end. Project 4 was originally led by Barry Scott before he retired, and then taken over by Murray Cox. And this was the end, well, I say the end, the end of the BPRC involvement with the grass endophytes, the epicloes, which is the backbone of the ryegrass industry in New Zealand. Almost every ryegrass seed sold in New Zealand has a beneficial endophyte inside, which helps against pest attack, disease attack, and drought. Since we started as a core back in 2003, we have had a strong emphasis on epicloe and trying to understand the fundamentals of how a grass inside, a fungus inside a grass communicates with itself and with the grass, and how does it actually do this. So this team have been able to get to some really, really deep science and really amazing stuff. So they understand the gene pathways that the fungus is using to produce the chemicals that actually give the beneficial effect. But some even more interesting work was what they were doing around the nucleus, looking at not just the genome but the structure of the chromosomes, and showing that where chromosomes actually touch is not random and drives gene expression. And so here's a whole new level. You know, we have linear DNA that we look at on the screen. That's not always going to tell you what the outcomes of that genome is because of the touching of chromosomes and the genes. So they've got a long way of this. It's quite an amazing piece of work at the fundamental end. So these two discoveries, the key pathways analysis and the structure of nuclei, help a lot in where you go next and how to make better strains for colonising grasses, and not just ryegrass. The team actually made a lot more progress than they had thought at the start of this project, and I would say that was true for all our projects. When we set them up, they were very risky, and everyone has delivered. And the work has been widely cited and has become a model system for other people working in these areas, and so huge impacts internationally. Project 5 was led by myself and Mark Hurst. This is about trying to enhance microbial-based biocontrol, in this case of grass grub, which is a scarab pest of grasses in New Zealand, with two bacteria that are known to cause a disease, and there is a commercial product out there and another couple coming on based on these bacteria for control. But there's an awful lot we don't know about these bacteria. And this project was around how do bacteria interact in soil when you have a whole lot of different strains and a whole lot of variability. And in this case, the disease is mainly encoded by a plasmid, an extra chromosomal piece of DNA that can, in theory, move between strains and not in strains. Half the strains in the field don't have the plasmid and don't cause disease. So what does that mean for the ecology of the disease? So we had 3 PhD students work on this. And the first one looked at the variation in the plasmid itself and tried to understand the evolution of the plasmid, where it's come from, where it's going. A little diagram down the bottom there, which don't worry, I'm not expecting you to see, is actually the evolution of the plasmid over time. And it comes back to what you saw in what I talked about in Project 3, mobile genetic elements. The plasmid is basically a construct of bits of DNA that come in and go out and can do all sorts of weird and wonderful things, including grass grub disease. But there are strong constraints we found on the plasmid moving between strains. So it's not promiscuous. It looks like it should be, but in the lab you can get it to go in these other strains, but we found that gene expressions changed, it wasn't as effective, or in some cases it just wouldn't happen. So the implications of this, there's some other work I haven't talked about, basically show that the presence of these non-pathogenic strains in the soil reduces the effectiveness of the applied biocontrol agents. So you have to factor in these non-pathogenic loadings in the soil when you're thinking about applying a biocontrol agent. And there are ways to improve the overall effectiveness taking this into account. Project 6 was our most ambitious project led by Ian Dickey, and most ambitious in terms of the scope. This was our first effort to start to look and say, what happens at an ecosystem level? You know, we're quite good at this biopesticide stuff, we're quite good at this classical biocontrol. What actually happens in these different environments? And they took a few different ways of looking at this, including these rather large 180 mesocosms set up in here at Lincoln, which had home and away soils and different plant combinations of exotics and indigenous, different insects were put in there and left to run for 18 months. So it was a grand experiment and is still churning out a lot of data. There was also field work to complement this and some specific experiments in the field. So one of the interesting ones was this part of the study did not find support for the enemy release. If you remember with the DOC project, they found in one case, it was enemy release that was driving the success. At the ecosystem level, that wasn't supported. So the exotic plants had a stronger interaction with the herbivores and fungi, so they could actually tolerate more herbivores and more fungi. So you weren't seeing necessarily nothing attacking it. There were things attacking it, they were just better at coping. So the land use change, where you just change a forest into a pasture or disturb things, actually increases the diversity of the plant pathogens, for example, and that's driven by the plant species composition increasing. So you're getting more plant pathogens operating in these environments. And the exotic plants showed a higher growth rate. So they were surviving these attacks much better than the natives. And then there's a spillover effect that the pathogens are building up on these exotics and then killing the natives. So there's a lot more in this project especially. It was quite a large effort. But so it basically shows the outcomes of this, that the movement of species, the movement of plant species especially, has resulted in a stronger and more variable interaction rather than interaction loss. So there's more things going on rather than less. So it doesn't become a simpler system. And the stronger species in turn are critical for the plant invasion success. And so they're driving what's happening in those environments. So I hope I've done justice to that. Project 7, firstly led by Leo Condron and then taken over by Amanda Black, was a specific iwi interaction project around carry dieback. And it was around what's facilitating carry dieback, the Phytophthora spread. So there's only about 1% of the original carry forest left. So carry are iconic species. We've got to save them. So the project was looking at how it spreads. One of the most interesting findings early on was that pines and grasses are acting as reservoirs for Phytophthora. So they're not acting as a break, they're actually it's where Phytophthora was being found in higher numbers than in natural carry soils. So one of the drivers of this they found was that the microbial communities under pines was different to the communities under old kauri forests, and especially different around some of the beneficial species we know, like some of the Pseudomonas, like the trichodermas, were less under these pines than you'd see in the kauri forests. And so there is an element of suppressive soils that's going on that means the natural soils are reducing the impacts of Phytophthora compared to these other environments. There's also a significant difference between the communities, the general communities of fungi and bacteria around asymptomatic kauri trees compared to symptomatic kauri trees. So, this obviously points to a promising direction for future reduction of effect, looking at a suppressive soils approach. It also highlights the importance of disturbance, and so destroying kauri forests and trying to plant them back into other environments is going to have problems. There was also a significance in the composition abundance of microbial genes related to carbon and nutrient cycling. So this is when you look to see how much carbon and nitrogen recycling is going on in these soils, and that's obviously reduced in some of these disturbed soils. Just quickly, when we first put the program together, we had the idea that we would run a Maori lens over a lot of what we did. I would say we were marginally successful at that. Two of our projects, Project 3 and Project 7, had very strong iwi interactions. But we did have a few wins. We had a number of Maori postgraduates trained during the 5 years. We helped support the launch of the Maori Biosecurity Network. They say success has a lot of parents, so we're just one of the people who put our hands up as helping. There were a number of others. Obviously the Kauri Dieback and PSA projects had iwi links. There was also an aligned project that Amanda did on re-indigenising the biosecurity system. But probably one of our most lasting effects was we assembled a very strong kahui led by Matua Henare, and that has led to where we're going with the new program, which we'll describe later. But it's probably one of our more lasting effects is actually to get the right people around the table. So obviously, as a Centre of Research Excellence, we like to tout our publications, and one of the things that I am very pleased about as director is not that we publish more papers, but we published in higher impact journals year on year. And so the quality of the research in the traditional metrics went up year on year. We also, since our inception, we've published over 1,600 papers. It's always a bit weird quoting these figures because no one exactly does research just on one project. There's usually a bit of mixing and matching, so we all claim the same things. But we have supported over 1,600 publications. A bit of a snapshot what a year would look like for us. 2018, 162 news and media articles, 118 seminars, 8 book chapters, co-authored from 30 countries on over 100 publications. So it's the quality of our research that really is important, but it's also being involved in national commentaries. It's having the team that is trying to influence policy and trying to influence thought, because that's what we should be doing as a Centre of Research Excellence. Unfortunately, this year, we've lost 2 of our alumni. Distinguished Professor Steve Ratton passed away after a very short illness. Steve was one of the original principal investigators in the centre back in 2003, so he's missed. Pierre Dupont was actually a postdoc of ours and spent a lot of time in the centre at Massey University, and he passed away in April as well, so it was very sad. Of course, on the other end, we have a lot of bright young things that we've cast out into the world. And this is really one of our major effects. So we can do a lot of really good research, but it's actually the people we train and what they can go do it later that will make the lasting difference in a lot of ways. And one of the things that we're really proud of is that they go into a variety of areas. We've had something like 9 of our graduates in this last 5 years go into MPI. But we also have them go into the universities, we have them go into the CRIs, and we will have one of our alumni speaking this morning who started off as a postdoc with us and went on to a career in plant and food. But we also have people who go into industry. So it's a really good mix of outcomes for us. And just finally, launching into the future. In 7 days' time, I think it's 7 days' time, we are launching Bioprotection Aotearoa, which has 7.5 years of core funding. Amanda and I will co-lead to start. And this is a real redesign of the program. This is not just business as usual. It took a lot of thought around what should we be working on. And it's around ecosystems. It's around getting away from trying to control one pest at a time, and how do you make more resilient ecosystems. And Amanda has led a group of PO leaders, so the POs are how we will break the research up, to come up with a program that integrates a whole lot of science to try and do the really gnarly question of what makes a healthy ecosystem and how do you improve it. We also will have 11 partner organisations for the first time. And so it's really pleasing because it means we're reaching into a lot of the science in New Zealand, not just the science that we're funding. Right. And the basic way to look at this, and there'll be a lot more over these 2 days about Bioprotection Aotearoa, but the 3 main areas are going to be: how do you define a healthy ecosystem? What tools can we develop to help defend that ecosystem? And then how do you design resilient ecosystems? And so that's the high level picture of where it'll be going. And look at that, I'm actually on time. So thank you. We're going to do questions at the end of this session, so please think about them during the session. And thank you very much. Now, it's my pleasure, as the chair of the session as well, to introduce Michelle Sands. Now, somehow someone has pinched my notes, but Michelle is the Environment Officer for HortNZ and has very bravely stepped in at the last minute because Nadine Tunley, who was going to give the talk, is the CEO of HortNZ, and she had to go to a board meeting. And so Michelle knows very little about us and has been very good at stepping up in the last 2 days. So I will let you do more of an introduction, seeing I pinched my notes.
My pleasure. Kia ora everyone. As Travis described, I'm the Environment Policy Manager at Horticulture New Zealand, and my background is in science, primarily in hydrology and water quality and catchment scale kind of natural resource management, but now I work in policy, and my areas cover climate change, air, land and water. I don't know much about bioprotection, and so what I'm going to speak to you guys about today is about the horticulture sector in New Zealand, but also about the vision of Horticulture New Zealand, which is healthy food for all, forever. And I'm going to speak about that from my perspective, which is in around environment policy, but perhaps that will offer some thoughts for your own work. There are 6,000 growers in New Zealand, 60,000 people are employed in the horticulture industry. The industry value is 6.8 billion, and 4.6 billion of that is related to export. And that means that about nearly a third of the value is related to the domestic market. We have 130 overseas markets, and growers produce more than 100 types of fruits and vegetables. And when we think about the very wide range of fruits and vegetables that are grown, some of them are grown predominantly for export markets. You know, our key export crops are kiwi fruit and apples. I think onions is next, but then there are very, very many crops, and some of those, a small proportion, is exported, and the remaining part is for domestic, and there are many crops that are grown solely for New Zealand consumers. So horticulture is a sector worth protecting, and as I described earlier, I'm going to help explain why I think that is. So HortNZ's vision is healthy food for all, forever. Oranga kai, oranga tangata, haere ake nai. And because I work in policy, I thought that it would be useful to define the terms. So that is what I thought I would do. So first of all, when we think about what is healthy food, I thought, well, it's more perhaps than just the body. It's about our minds and our as well. And so, of course, people like the idea of eating healthy food. And so, as an industry, we tap into that and we promote the health benefits of eating food. And so, for example, here's a Zespri, marketing which talks about immunity. What's really important underpinning those claims is the research that supports, the science that supports those nutritional claims, so they are robust. The other aspect is around certification, so people want to have reassurance that the food they eat is safe. The GAP programs are really important, and all growers in New Zealand who sell to a supermarket or export have to have their food certified by independent assurance. And those assurance programs look at a range of elements, but including food safety. And so that, would look at key aspects such as, agrochemicals and, or water that might include E. coli or compost, and to ensure that the food that leaves those growers is safe to eat. What we have at the moment in terms of environmental management is a focus on a kind of integrated farm planning approach, which is looking at adding more elements to farm plans. And so I work in freshwater, where we're expecting, freshwater farm plans to become more common, in greenhouse gas management, but also into biodiversity and biosecurity. One of the things that we at HNZ advocate for is that those sort of integrated farm plans should be managed within a common assurance framework, and that assurance framework must be robust, because the claims that we make about the health of the food that we eat, or the safety of that, or the degree of sustainability, it must be trustworthy. The other aspect, of course, about healthy food is actually its health for our bodies. And so, I think the Ministry of Health recommends that people eat 5+ a day. In New Zealand, low fruit and vegetable consumption and high sodium intake was the second leading cause of health loss in New Zealand. So it's not just about the idea of healthy food. People actually need to eat healthy food too. And at this level, it's not just about the value of selling products to people and the benefit to the economy from that. It's also about the health cost of people not having the nutrition that their bodies need. Of course, the other part of healthy food as well is perhaps about our soul, the way that food makes us feel. And I think in all cultures, the eating food with your friends and family, the ability to share food generously and receive it with grace is very important for people's well-being. It's also about the role that food plays in meeting people's cultural needs. And, in New Zealand, we have a wonderful climate and the ability to grow a great many crops. And we have a history of growers from different parts of the world who have brought different growing traditions to New Zealand. For example, the Chinese growers who have been so important in providing green vegetables for New Zealanders over a long period of time, and a great range of vegetables. Those growing systems are at risk now because they tend to be of a small scale and in the peri-urban areas where the land value is very high and where there's increasing pressure for urban development. The other part, of course, about food is about the food that we don't eat, or the food that is impacted. And I have a picture up here of inanga. So, in New Zealand, many of our lowland streams have been drained. The habitats that would support mahinga kai have been compromised by the choices that we have made about the food that we are producing, the food that we produce for export and the food that we grow here. And so that has an impact on the choices that people can make. And then also the ability to grow your own food. There is a real connection, and particularly I think for urban people, a kind of a loss of sense of place. And so the role that growing your own food in a kind of a community scale can have in terms of people's well-being is important. Of course, for HNZ, our vision is about healthy food for all, forever. And so the scale is larger than just that sort of community scale. When I thought about what the term "for all" could mean, I looked to the Fit for the Better World vision, which is a sort of a strategy developed by the Primary Sector Council, and it has this statement, and it says, "We are the farmers, growers, fishers, makers and crafters of Aotearoa, New Zealand. We aspire to an enriched future by providing the world's most discerning consumers with outstanding, ethically produced food, natural fibres, drinks, and bioproducts, all sourced from our land and oceans." And it also has this kind of principle of taio ora, tangata ora. If the natural world is healthy, so too are the people. And I think that the taio ora, tangata ora principle resonates with me, because my way of thinking is that, people are part of the natural world, and so what is good for our bodies is likely to be good for the world, and vice versa. And what this vision kind of, I think, looks towards is a place where our food system in New Zealand is in more balance with our natural world, and we're able to gain higher value from our products and sell those to the most discerning consumers. And you know, in horticulture, we do sell high-value products. Kiwi fruit and apples are premium products that are exported throughout the world. And the interesting thing about those products is that they're not premium because they are value-added, really, or processed. They're premium because you can eat them raw. You know, you can eat them without anything. It's something about the simpleness of them that creates their premium value. But there perhaps are other opportunities for new crops or nutraceuticals that we could sell, you know, throughout the world that we could produce sustainably in New Zealand. However, For All isn't just about selling, you know, those premium products across the world, because as I described earlier, in the horticulture sector, we have a strong part of our industry is focused domestically. And because of New Zealand's geographic isolation, we can't look to import fresh, you know, all of the fresh food that we need to eat here. We are reliant on growing it. And so, you know, if growers were to produce that 5+ a day that we need, you know, that would be, you know, 15 million servings of vegetables every day, 10 million servings of fruit. It's a lot of food that needs to be produced for New Zealand here. Unfortunately, you know, New Zealanders are not eating 5+ a day, and only about half of us are eating 3+ vegetables a day, and it is declining year on year. And interestingly, it's declining at about the same rate as our vegetable growing areas are also declining. There's been a recent study looking at food hardship and early childhood nutrition, and 1 in 5 children in New Zealand live in food insecurity. And this is a recent study that was done by Bruce Kidd, I think his name is, and others, and it looked at the cost of a healthy diet versus the kind of current diet in New Zealand. And this is an interesting study because we've seen quite a few of these sorts of studies done internationally, looking at what is the diet that would be more aligned with, you know, a low emissions future, or, you know, more aligned with, you know, being good for our bodies, but also within environmental limits. And what we find is that a healthy diet is more expensive than the current diet that New Zealanders eat. And when we go further along and look to the diets that are more likely to be aligned with a low emissions future, they are the most expensive. And so this is kind of a challenge in terms of how do we, you know, produce food, how do we meet all of our kind of the costs associated with producing food in a way that is sustainable, but also produce food that is reasonably priced so it can be accessed by all people. And, you know, this is a challenge because fruit and vegetables in New Zealand are, you know, are still actually quite cheap, really. And many growers would say that they couldn't, you know, they can't grow them, you know, for a lesser price. So there is a difficult challenge here, and it's a societal one. But one of the things that I will come back to is thinking about, you know, how we manage our natural resources and the way in which we provide for things that are essential for human health. So the other aspect of our vision, healthy food for all, is forever. And so forever, you know, it's a long time. Maybe it goes back as well as forward. And when we think about forever, I think at the heart of that is sustainability. And, you know, when we look to the future in New Zealand, we have to think about a future which has, where climate change is part of our future. And so part of my work, I work in, you know, looking at how the agriculture sector can reduce its emissions. I'm on the steering group of Aotearoa. And half of New Zealand's emissions are related to animal agriculture. And we know that there is an opportunity for, you know, the diversification of our farming systems into more horticultural products to reduce emissions. And of course that would need to be market-led because you have to sell the product. So that's something that we would have to grow before we could really expand. But we also need to think about the future challenges as well. and the resource needs of horticulture. We do need, we need highly productive land to grow on. We need reliable water. And so, what are the challenges that are coming in the future, and including, in the context of climate change adaptation? So, one of the things that we might expect more in the future are more extreme events. And so, one of the ways that we might, one of the ways we might protect ourselves from more extreme events are things like, crop protection structures. But we also see, Horticulture New Zealand has been involved in environment court cases where parties have objected to crop protection structures on the basis of amenity. They don't like the way they look. And so, we have challenges in terms of adapting our landscape so it is more resilient to a future. Another aspect here, around, the risks that we have already in terms of biosecurity and the potential for that risk to increase with a changing climate. One of the ways that growers manage this, vegetable growers manage, pest and diseases through crop rotation. And this is, really important, and all, outdoor vegetable growers in New Zealand would rotate their crops. But we struggle in terms of environmental regulation because we have environmental regulation in New Zealand which is really designed, I guess it's the 80-20 rule, it's designed for pasture farming. And we have struggled to get policy that would recognise and provide for crop rotation. Just this week, NZ is going to join an Environment Court appeal where parties have appealed the definition that would provide for crop rotation. So whilst we hear about regenerative farming, there are barriers, barriers that are, it's hard to understand exactly what the motivation is, but I think it's related to the ease of administrating things if they are sort of fixed in space rather than sort of moving about. And the final picture here is really about water. Horticultural products rely on a reliable supply of water. We know that our freshwater receiving environments in New Zealand are under pressure. Many of them are over-allocated in some part in terms of their water quality or some parts of their flow regime. And so we need to think about how we manage our water resources differently and more carefully in the future. There are many opportunities to increase efficiency. There are opportunities to store water and augment water, but we also need to think about the functioning of those rivers and streams and how we can provide for those in the future. And the other part, I suppose, this here is, I won't read this, but this is part 2 of the RMA. So the concept of sustainable management, it's not a new concept. I've been working in the RMA for more than 20 years, and in the RMA we had this concept about future generations. We understood about the life-supporting capacity of ecosystems, and we understood that the way and the rate that we met our social, economic and cultural needs would need to be adjusted to provide for those other parts. But we didn't necessarily achieve that, and I think one of the weaknesses that we had was, the assessment of cumulative effects. And that really is about the question that we asked. So whilst we sought to assess environmental effects, we weren't very good at assessing cumulative effects. We are now coming into a new time in resource management. We have, new acts that are being developed now with new concepts to help us hopefully develop a food system that operates within environmental limits and with a changing climate. One of those concepts that I'm familiar with is Te Mana o Te Wai within the National Policy Statement for Fresh Water. And there are a number of elements to that concept, and including a greater role for Māori in the management of water. But also it includes a hierarchy of obligations, and that puts the mana of water first and the essential health needs of people second and everything else third. And I think that that is a useful framework in helping us, the part that we struggled with in the RMA was how to achieve the well-being of people and the well-being of the environment, whereas some places and sometimes they seem to be, in conflict. One of the arguments that NZ makes is that domestic food supply is an essential human health need and should be provided for within our natural resource management allocation frameworks. The other concept that is now being talked about and that we anticipate coming through into the new legislation is te mana o te taio. I won't talk about that because I don't quite know what form that will take yet. So I suppose, just summing up really, I was thinking about, well, you know, I think we're moving from a time when we operated in the RMA, we hoped that our decisions were sort of agnostic. But when we look back at the decisions that were made, you know, maybe they weren't. And so now we've come to a place where our environmental limits are reached, sometimes overshot. And so we are becoming more explicit about our values and decision making in order to determine how we can claw back natural resources and provide, you know, for a future that is more within environmental limits. So my question really was about, you know, the role of science in that. And I think there is a real need for science, you know, for scientists to have the courage to consider problems and to be open to being wrong. You know, that is such a powerful way of, you know, methodology for assessing problems. And the other aspect of science, which is really important in our future decision making, is to have the courage to speak the truth. And, you know, and I understand it does depend on the question, and, you know, you can only speak the truth as you understand it, but it is very important. And to speak that to decision makers, but to everybody, because we're in a time now of so much information, but, you know, not all of them, you know, so much of that information might be, you know, nonsense. So I thought I would leave you with a, you know, equally lofty vision, seeking truth for all, forever. Thank you for your work.
Thanks, Michelle. As I said, we will have a question session at the end. So, seeking truth for all, you know, just a small vision. All right, so next it's my pleasure to introduce Jay Sarama. And Jay is one of our success stories. So he started as a postdoc on Project 3 and has been taken on in a full-time position in plant and food research, which is exactly the model we like to see. So, Jay, I will turn over to you.
Thanks, Travis, for that. Short intro. Kia ora koutou, everyone. My name is Jay. My pronouns are he, him. So as Travis has already introduced me, I'm a scientist working for plant and food research. So today I wanted to share with you some of the lessons that we learned as a result of some funding that we got for a postdoctoral position at Plant and Food Research, working on, title gives it away, PSA. And some of the lessons that we learned, some of the learnings that we've, and technology transfers that we achieved as a result of that funding. I apologize in advance that this is a very science-heavy presentation. I welcome any questions after. So I'll jump straight into it. So PSA. So PSA, Pseudomonas syringae pathovirus. Oh, okay, all right. Pathovirus is a pathovirus of Pseudomonas syringae, a bacterial pathogen. You may all be well aware of this by now. Caused a devastating disease, bacterial canker disease in the kiwi food industry, completely destroyed the current, then current gold cultivar Hort 16A, and some of the symptoms are on the screen there. And so the PSA itself, pathovirus, is divided into multiple clades. The one that came into New Zealand in 2010 is PSA 3. And it's the most virulent of the lot. But there are other clades that are out there, PSA 2 and PSA 1. It's also related to a mild pathogen of kiwi fruit that's ubiquitous around the world, PFM. I won't be really talking about that at this point. Pictured on the right there, bottom right, are Hort 16A plants that we use in tissue culture. So these are plants that when we infect with PSA, after 50 days, these are the typical symptoms that you see. The symptoms kind of develop progressively up to 30 days, and then there's a rapid sort of switch to the plant dying and completely losing control. So we wanted to understand what the mechanism was behind this disease, and so this is a very critical fundamental research. There's a lot of research that has already gone into Pseudomonas syringae, less so at that point when I entered into PSA and its mechanisms. So, oops, sorry. So Pseudomonas syringae, on her journey of self-discovery, history of a pathogen, becoming all that she can be, we sort of look at what are the things that are essential for P. syringae to be able to be a pathogen. And so along this journey, I've indicated with a red star, is the acquisition of a very complicated name, a type 3 secretion system. It's basically just a molecular syringe, pictorially represented on the right there. So the blue lines are the membrane of the bacterial cell, and so what the type 3 secretion system is a set of genes that Pseudomonas syringae has acquired that allow it to build this protein system that can then inject proteins, other proteins, into the plant cell. So these proteins are effectors, as Travis has mentioned earlier. They are injected into the plant cell, and that performs a variety of functions. Through studying other similar Pseudomonas syringae pathogens, in this case tomato, the community, science community has identified several effectors that are key to causing disease. Most of these effectors are involved in suppressing the innate plant immunity. In plants, every cell is able to defend itself. It doesn't have a circulatory system like blood flowing through it the way that we do, and so it doesn't have a moving immune system flowing through it. So every cell defends itself. So it's, in a way, it's very good, because then every cell can then stand up for itself. But that means that a bacterium has to come in, and every bacterium has to defeat the system. And so it uses the type 3 secretion system, injects these effectors, and the vast majority of them are involved in immune suppression. A small handful, so in the blue at the bottom there, HOPM1 and AVRE, the names aren't that important at this point, but they're effectors that are involved in making a wet epiplast. So they release water from the cells into the empty space within the leaves, and this pulling out of the water allows nutrients to flow out, and this is absolutely essential for the bacterium to be able to grow, divide, and replicate. And so it became very clear early on that this is going to be very important for a Pseudomonas syringae pathogen. And so this is when we sort of entered with PSA. This was all knowledge that we had. So with PSA, it has a large number of effectors, quite unfortunately, distributed amongst the different biovars or different strains of PSA. So the one we're primarily interested in is PSA3. So the way, so when I came in, we were interested in knocking out, so genetically removing each of these, either in groups or individuals, of these effectors to understand what contribution they played in the disease of kiwi fruit. And so I'm not going to go into the details how we did the knockouts. I'm just going to hand wave and say we did a little bit of science magic, and we got a collection of these mutants. It took 3 years, but it was very rewarding, very easy for us. So we got a collection, we call it a library of knockout strains for individual effectors knocked out. And then we use a classic in vitro infection assay. It's basically those little plant tubs that I showed you earlier, a photo of. We, in a very complicated setup, we take the plants, we flood them with 1 million cells per mil of PSA, and then pour it out, and then wait, and sample the leaf tissue, grind it up, and look at how much bacteria is in there. And we also keep some plants left over for some photos, because everyone loves photos. So for the quantification, what we're looking at, I'll sort of depict it here. Travis put a similar version of this up earlier. So in every graph that I'll show you, the red bar represents the original PSA strain. So it has very high virulence, and that's depicted by the height of the bar. The higher the bar, the more PSA is colonizing the plant. And so the scale on the left is a log scale. So 2 is 10 times more than 1, 3 is 10 times more than 2. So it goes up very rapidly. This is exponential growth. So if you see a slight decrease between 9 and 8, that's 10-fold more. two slots down, it's 100-fold less. So what we're looking for is when we knock out a gene, in this case, the HARC-C gene. So this knocks out the entire secretion system because it loses one critical component in that injection mechanism. So when it loses it, it's completely unable to infect the plants. The plants are healthy. Photos don't really show it here, but they're largely healthy. And they're unable to colonize the plant. They sort of persist on the surfaces and on the inside, but they really aren't able to cause disease. So this was the very first step. This is all knowledge from other Pseudomonas syringae. We're just replicating it in the lab. So the first sort of thing we tried was knocking out that effector that's involved in sucking out nutrients from the leaves. So we knocked out AVR-E1, and very nicely we saw a strong decrease. It's a 1,000-fold decrease or more in its ability to colonize the plant. And associated with that was a loss in its ability to cause disease as well. So unable to colonize, unable to cause disease. Then we took the library that we already had of all these effector knockouts, and we screened them on kiwi fruit over a process of months. And we found 3 other effectors that are individually required quantitatively that contribute to colonization of PSA. So we found these 3. These, however, don't reduce the colonization enough to reduce symptomology. So you still get disease-like symptoms. So they have a quantitative contribution, but they can still cause a weaker form of disease. So obviously that's those 4 individually contributes. What about together? You know, effectors like working in groups, just like humans like working in groups, unfortunately. So we group them into what we call redundant effector groups, or REGs. So what we found is very nicely this large locus of 10 effectors on the bottom left there, those are the 10 effectors, that when you knock them all out together, you see a small reduction in virulence. When I say small, it's about 10-fold. So it is significant, but on the graph it looks small. So it's very important that we sort of understand collectively what contributes to immunity, into the causing the disease. So this reduction, again, doesn't cause enough of a reduction to remove symptomology, but there is a quantitative contribution. We lucked out because these were all sitting in what's known as the exchangeable effector locus, all 10 of them sitting right next to each other, making an easy target. We can just knock it all out all in one go, and so we understand it very nicely. However, a smaller subset, you can sort of see it labeled as the FEL, those 8 at the bottom. When you knock it out, it's still fully virulent. If you knock out the top 2, it's still fully virulent. You have to knock out all 10, or a subset of those 10, which we're starting to narrow down now, to get that small quantitative reduction in virulence. And then finally, another redundant effector group. There's a lot of work that went behind this. I'm not going to cover that because these are all in different locations throughout the PSA genome. So we had to go fishing using knowledge that we had from other pathogens and try and knock them out sequentially to figure out how are they contributing to virulence. And so we found another 5 vectors that work together as a group. So to summarize, 4 individually non-redundant effectors that contribute to virulence. One of them is a major contributor, AVR-E in red, and 2 redundant effector groups, REG1 with 10 effectors and REG2 with 5 effectors. What does this all mean? It's just numbers and names at this point. So the key about this is that we are learning so many things through this process. One of the earliest things that we learned was that effectors so before this, there were a handful of other pathogens that had been characterized similarly in sort of a sporadic way. But this work, funding over 3 years, allowed us to sort of do it in a collected, really condensed, concentrated form on one pathogen. But interestingly, all the studies that have been done before were done on what are known as annual plants, so plants that have a lifetime that span usually a year, a season. And there are some studies on ones that are biennial, so over 2 seasons. But the problem with this is if you imagine a plant that has resistance genes, and we're talking about resistance here, where the plant's able to defend itself against a pathogen. For an annual plant, the resistance genes only need to get the plant over the line until it can reproduce, to flower, set seed, and sort of spread itself. So it has like a short window. It just needs to survive that gap. perennial plants, kiwi fruit, for example, the plants last for decades, 60, 70 years sometimes in orchards. So when this pathogen came into New Zealand and it destroyed the orchards, this was a huge tragic loss. These are growers that have plants in the orchards that they know. They're like family members, effectively. And so they go in and they see this tremendous loss to their orchards, plants that they've had for decades. It was a huge loss. But on the flip side of this, we know that, you know, there's kiwi fruit out there that is resistant to PSA. PSA is from China, where there's a variety of kiwi fruit species that are growing there, and there's a large variety of PSA3 growing there. And so some of these plants will have resistance genes that are durable, and we love that term, durable resistance, because it means that these resistance genes allow it to resist PSA and survive for decades. So that the lessons that we learn from this is essentially completely different. These are resistances that allow the plant to survive despite infection. So that's the first thing. It's that we understand the effectors have an additive role in virulence. This is something that we had hints of from other pathogens. We now have a model that we are developing further and further for a perennial plant host. And we have, so how do we then deploy it into resistance? So obviously these are virulence targets, effectors that target the plant and suppress its immunity. If we can identify how they're doing these things, we can try breeding out those susceptibility targets, because these plant proteins are Achilles heels for the plant. We can effectively take them out. We may be able to take them out, and so the effectors are then useless to the pathogen. And so if they lose these effectors, they'll automatically become less virulent. So it's a really durable system of breeding resistance into plants that hasn't really been deployed very well until, well, hopefully until now. The second thing that we were able to do was then use that knockout library, and this is in collaboration with some master's project funding that we got from Zespri. And this is work that a current PhD student, Lauren Hamera, is doing. She did it for her master's research in our lab. She deployed the same library and found 4 effectors that are recognized in a resistant variety of kiwi fruit. So this is kiwi berry. You may have seen it in supermarkets, spanning, I think it's only 2 weeks that they're available in supermarkets. But it's a small berry-sized fruit. And the plants happen to be resistant, completely resistant to PSA. So unlike the previous graphs where we're looking at a high red bar and it dropping, what we're looking for in this case is a low red bar increasing because it's losing resistance, and so it's growing a little bit more in kiwi fruit. So as a result of that earlier funding, we were able to deploy this and find really interesting targets that we can directly feed into breeding programs to introduce these resistance genes into new cultivars. And the second project that we were able to do was looking at the LPS. So the LPS in orange on the image on the left there is a extracellular glycopolysaccharide that's produced by PSA and a large number of gram-negative bacteria. We identified a locus that determines the shape of the LPS and its sensitivity to these toxins, talasins. They're like phage-derived molecules. And so as a result, we were able to publish a paper that outlines strategies to maybe use talasins as a treatment in orchards for management, because currently it's just managed with copper treatments, but you can use talasins. As well as some future phage work. So it allowed us to establish 2 collaborations, one with University of Auckland, another with University of Otago, that are ongoing to this day. So to sort of summarize, that should say Bioprotection Aotearoa, sorry. BA partnerships fundings means new science. So this is, we characterize a novel model pathogen and plant system, resistance breeding for new cultivars in the future, novel on-orchard control treatments, as well as the ongoing collaborations that we have now as a result of some funding that we got for the project very early in understanding PSA. Ngami.
Well, that brings us to morning tea. So the speakers, thank you very much for staying on time. And we will be back in here. I'm starting it at exactly 11:10. So tea and coffee are just out in the foyer. Thank you. We've got a session of questions at the end. Don't go far, Peter.
I just saw gesticulation before.
Okay, everyone, we'll make a start, please. So we have one more presentation in this session, and then we will have question and answers from all the speakers up the front. So it's my pleasure to introduce Professor Peter Dearden, Professor of Genetics at Aotearoa University and also the Director of Genomics Aotearoa, and been a member of the research team of Bioprotection Research Centre for the last 5 years. So, Peter.
Hello, Peter Dearden, how? Real delighted to be here. Sorry, running out of words. There are so many of you. It's a real delight to be here. Thank you so much for inviting me. I'm going to talk to you about some stuff, and I know that I had a I pointed it the right way. I know I had a title in the thing about something else. We'll get to pest control, but I wanted to talk a little bit about weevils because they have really cool noses. So, unfortunately, Travis has said everything that I'm going to say in the start of my talk. No, so I want to talk about what we've been doing as part of Project 2. There's our big Project 2 team who've been doing stuff for the Bioprotection Research Centre, and how that has sort of informed my thinking about what we need to do in the future. So we will talk about pest control. I'm not going to talk about gene drive specifically, but I'm going to talk about what I think our challenges are, and it's going to get a bit opinionated and shouty, okay, so just strap in. Sorry. We've already kind of introduced to this guy. This is, got a pointy thing here. I do have a laser, there we go. So this is the Argentine stem weevil, which is, I must admit, my favorite stem weevil. It's a pest. It's a pest of grass. In particular, its larvae burrow into the grass and they cause damage to grass. And, you know, what New Zealand sells effectively to the rest of the world is grass. So we have a problem here. This affects the basis of a lot of our pastoral industry. So that's a big issue. And, of course, it causes pasture damage like this. This is not tasty for sheep or cows. This is Argentine stem weevil territory. So stem weevils are a problem, and they've been a problem for some years, and now it's not working. Come on, you know you want to. There we go. Stem weevils were first recorded in New Zealand in 1927. When we assume that they came in from forage being brought from South America, but it's not clear who, why, where. You know, just appeared, well, first recorded in 1927. Costs about $200 million a year, but that's the cost currently. It's controlled by something we're going to talk about in a moment, and so this is the cost after all of our control measures. It's hard to kill with insecticides because it's encapsulated in the leaves at its sensitive stages, so if you wanted to use insecticides to kill stem weevils, you have to treat with a hell of a lot of insecticide. It's also affected by endophytes in the grass, but endophytes obviously, the endophytes that are effective against this weevil often cause ryegrass draggers in stock as well, so there's a sort of balancing thing here. So it's a problem. It was a problem that we thought, well, at least Stephen Goldson thought we had solved by the introduction of a parasitoid wasp, Microtonus hyperidae, which was brought from Argentina and released between 1989 and 1990 around New Zealand. And remarkably, this turns out to be one of the most effective biocontrol programs in the world. When this wasp was first released, let's have a look at the wasp. There he is, or she is. We got rates of up to 80% parasitism. So 80% of the stem weevils in pastures were carrying around a larva of this parasitoid wasp. And what happens is the parasitoid lays its eggs in the weevil, the egg hatches into a larva, the larva dangles about, eats the weevil's insides, and finally hatches out, killing the weevil. Right? So 80% parasitism means our populations of stem weevils were crashing. So that's great news. Wonderful. And biocontrol systems are they distribute themselves, the wasp goes everywhere, and it's self-sustaining. It's wonderful. We've solved this particular problem. The issue is that we haven't. And if the next slide, there we go. What has actually been seen, I've messed up my slide somehow. What you see in these graphs from a paper which you can't read, is that there's been a steady decline of this biocontrol over time in lots of different areas. Down from, if you look at this Waikato where we've got great data, you've got like 80% parasitism and it's now dropped to sort of below 20%. That's over a considerable period of time, but this is a huge difference in the number of Argentine stem weevils in our pastures and the damage that's going to be caused and the control measures that need to be done. This is a massive cost. As Travis told you, do I just point at you? There we go. One of the jobs, one of the things that Project 2 did in the Bioprotection Research Centre is monitor Argentine stem weevil populations up and down the country. There's a massive number of sites that were monitored, and the outcome of that is that if you look at all of those sites, there has been a parasitism decline from 70% or above down to 10% and above, and that decline is really related to the years after the first parasitoid release. So you release the wasp, and you've got 24 years of reasonable biocontrol. But then it's game over. So that's really unusual, and certainly unusual for a biocontrol system. Why this would happen makes not a lot of sense. And so one of the jobs of Project 2 was to try and understand what was going on here. And the problem is, to be blunt, what we have here is a pest that was, came to New Zealand. Nobody really knows anything about it because nobody cares in Argentina. And a parasitoid wasp that I have reliably been informed Stephen Goldson found on the lawn of a hotel. Brought to New Zealand, very effective biocontrol, but actually we know little about it. There's been lots of work on looking at how it might parasitize, what other off-target effects there are, but it is a system which is working, and therefore we've kind of said, well, it's fine, we'll be all right. So Project 2 and a whole load of other people around the country have been trying to work out what's responsible for this decline. And it's something to do with me pointing in the wrong direction. So one thing we thought, instantly being geneticists as well, obviously if we've got a stem weevil population and a parasitoid that comes in and starts killing that stem weevil, then we should see, if those stem weevils are becoming resistant to the parasitoid, we should see strong selection on stem weevils, right? Only those stem weevils which are resistant should survive, so we should see that in the genome. So we looked at whole genomes, or we sequenced the whole genome of the Argentine stem weevil, which turns out to be big and ugly, and we did genotype by sequencing of stem weevils from up and down the country, and the answer is we can't find a clear sign of a resistance locus. There is signs of genetic variation in the population, so there's a north-south cline. Weevils in the north of New Zealand are very different to weevils in the south of New Zealand, and there's a massive amount of variation. Not consistent with stem weevils came into the country once and spread throughout New Zealand. More consistent, in fact, with our modelling, suggests multiple introductions of this beast. We've got stem weevils, they have a lot of genetic variation. So that didn't help. It was expensive, it didn't help. So I think that's the motto of genomics. Genomics, expensive and doesn't help. It's probably the wrong thing to say at this point. Okay, let's think about COVID. It's really helped with COVID, all right? Sit down. Okay. We also thought, okay, well, let's try and understand what happens when a stem weevil is parasitized. So we have systems where we can look at gene expression. Oh, now we've gone past. Okay. We can look at gene. Can I do it with a keyboard? No, okay. I'll just wave at you, shall I? So this is a complex slide, and we're not going to go through it in great detail, but we can look at variation in gene expression between weevils that are parasitized and not parasitized. Weevils we think that are that are resistant, and weevils we don't think are infected. And the fact of the matter is we can find genes that are associated with these particular traits, right? So the genes that are upregulated in weevils that are resistant. What those genes mean is really hard to do, and I really want to thank the last speaker who talked a lot about taking genes and functionally testing them. And that's the most important thing here. So in a culture system with bacteria, you can do that. In a Argentine stem weevil, we cannot do this. We don't have permission to do it. We don't have the ability to, well, we probably could do it if we tried hard enough, but we would need containment facilities, which we don't have. So we can look at these genes. We found genes that look to be associated with resistant or genes that are parasitized. We can even look at the genes that are expressed by the embryo of the parasitoid in the wasp. So we've got some really cool data, and this is the work of Sarah Inwood. Also in here, beautiful timing, we have found a virus. So a sort of unknown virus. There's been work on viruses in some other parasitoids, and viruses are often in parasitoids and are involved in affecting the host, but we also found an unknown virus. This virus, now you did the thing. So this virus has, so in Microtonus hoperoidae, which attacks the stem weevil, we've got this virus, and actually the virus that it's related to, there is only one of. So this is a kind of a new clade of viruses we haven't heard of before. We know it's a virus through complicated stuff, looking at read depth of the genomes and things. We know that this is actually an infection that these wasps carry, and they transmit it onto the weevils. So this may be something to do with that parasitism role, but there's something interesting happening here we didn't know about. But I haven't told you the smoking gun, right? There is no smoking gun here. What we haven't found is being able to say, look, here is why resistance is occurring. Okay? We have a general idea, and that's something that Travis talked about as well, and that is that the stem weevil is sexually reproducing while this parasitoid which attacks it isn't. Okay? So this is a problem, and you think about this, if a species is sexually reproducing, it's able to recombine its genome and produce variation, and that variation is able to be selected for. But the parasitoid attacking it is asexual, and therefore it's producing clones of itself. So it's very hard to see how it can change greatly over time. And awesome modelling work from Paula, Stephen, and Jason have shown that actually if you look at parasitism rate, if you model parasitism rates against sexual parasitoids, so we've got a sexual host with a sexual parasitoid or an asexual parasitoid, you can see within the asexual parasitoid, we predict that this will drop off over time. Something we never thought of before, but actually it's quite clear that this is a serious flaw in the way that we have thought about releasing parasitoids for biocontrol. So we have to ask the question, what the hell do we do about it? So we know that in New Zealand there are actually a bunch of weevils which are controlled by parasitoid wasps. So there's the Argentine stem weevil controlled by Microtonus hoperoidae. There's the Lucerne weevil, which is also sexually reproducing. It's controlled by Microtonus ethiopoides, a sexual strain from Morocco. It's, I don't know, Morocco always seems like more sexual than... Ethiopoides, the same species, another strain, an asexual strain from Ireland, not surprised, controls the clover root weevil. And finally, we have a French strain, which is sexual, which also attacks the clover root weevil, but we don't use for biocontrol. And you can see that we've got some problems here. Argentine stem weevil, that's $200 million a year at the moment. Clover root weevil is $220 million a year at the moment. Clover root weevil is a massive problem. If this is released from its biocontrol, I think we're going to see major effects on our pastoral industries. Has an asexual thing. So we would predict at this point that we're going to lose control of the clover root weevil. So we thought to ourselves, is it possible that we could take some of these wasps, and as you see here, this species, Microtonus ethiopoides, has different strains which are sexual and asexual. So these are probably quite recently asexual. Is it possible for us to switch them back to sexuality? Can we understand how they're asexual and make them sexual again? Microtonus Aotearoa is probably sexual in its native range. It's just that Stephen chose an asexual one. Say no more. What we've done is we've sequenced the genomes of all of these guys here. We've sequenced the genome of the stem weevil. With Neil Gemmell, we've sequenced the genome of the lucerne weevil and the clover root weevil. And so we have genomes now of all of these species. Some of those genomes, let's go for slides, let's just go fast. So some of these genomes we've used Hi-C, so we've got chromosome level assemblies of some of these wasps. Just keep pressing the button. We've looked, and particularly trying to look at genes associated with meiosis. So meiosis is the thing that does sexual reproduction. It's the form of cell division. So we were interested to see whether genes associated with meiosis were affected in strains that were asexual. And the answer was no, but yeah, keep going. I was going to point, but we don't need to. We've also looked at olfactory receptors as a way to think about how these wasps are finding their hosts. Clearly, there are different strains of Microtonus ethiopoides which attack different weevils. How do they know? We reckon it's through smell, and actually there's a little clade. I'm going to point to the thingy. There's a little clade here. We found a whole set of olfactory receptors which are specific to this clade of parasitoid wasps, which we think might be involved in the host control. But of course, I'm not allowed to knock them out and see whether my wasps now can't find their host, right? So we have this problem that you bacterial guys don't. Okay. The other thing we've discovered, and this is a completely uninterpretable picture for most of you, but this is actually the ovary of Microtonus ethiopoides, a Irish asexual strain, stained for a gene which is involved, stained for a protein which is involved in meiosis, and you can see this staining up here, these little cells. These are cells undergoing meiosis. So, in fact, meiosis is unaffected. These guys are asexual, but they're doing meiosis anyway. So there are mechanisms why this might be occurring, but it suggests particular routes to asexuality. We know what genes are involved in those routes. We have some ideas of cell signaling pathways which seem to differ between these things. In the lab, we can change some of those genes, and we have seen some signs of producing embryos of different sex, but they're in the lab manipulations. And the techniques we're using are not things we can do outside of the laboratory. So it looks like we're getting to the point where we even might be able to change the switch back to sexuality in these species, but we're doing it with technologies that are not applicable on a broad scale, and that's our problem. Someone's messed up all my slides. I blame you. On the other hand, I don't blame you because you're changing the slides. So it's a great guy. So the other thing that we're thinking about is saying, well, are there ways we could make these wasps more effective? There's some wonderful work out of Australia that you'll be seeing in the media quite a lot recently, where they've manipulated the microbiome of dengue fever-carrying mosquitoes, and in doing so, they've blocked the transmission of dengue fever in those mosquitoes. Those mosquitoes are being released in Southeast Asia, and we've shown just a decrease in rates of dengue fever in areas where those mosquitoes have been released. This is not genetic modification, but it is messing around with stuff. So we're planning in the new Bioprotection Aotearoa to do the same thing, looking at a particular problem in honeybees, which is deformed wing virus transmitted by the varroa mite. Can we block transmission of the deformed wing virus by messing with the microbiome? And the same applies with our parasitoid wasps. Can we mess with the microbiome and improve transmission of that novel virus we found? Can we cause a weevil pandemic and wipe the bastards out? This is probably not the right time to be talking about weaponizing viruses. But I do want to say, look, joking aside, Our problem is our control of stem weevil is failing, and Project 2 has done an enormous amount of work to understand why. But all our solutions to the problem are problematic. And that's because they involve manipulating biology. And manipulating biology is hard and controversial. I think we have to think about this in terms of not just our primary production, but also our conservation estate. And I'm not going to read all this, but the headline is, we're not winning. Those species in New Zealand which are endangered are becoming more endangered. Our native ecosystems are becoming more endangered. Our primary production systems are becoming less productive. And all of this in the background of climate change, which we all go, oh, and climate change is coming. Well, it's not coming, it's here right now, and it is affecting all of our systems. Let's look at that in terms of a weevil. Oh, I'm pressing a button, but you're doing it for me. So this is my model of climate change. Like, it's going to get hotter, and that heat is going to move down the country. I know this is trivial and pointless, but Argentine stem weevils are distributed in the same way. Higher populations in the north, lower in the south. Our Argentine stem weevil populations are going to move south. Our resistance to the Argentine stem weevil is distributed in the same way. Resistance is higher in the north than in the south. So our resistant weevils are going to move south as well. So we're not just failing in our biocontrol, we're also going to have climate change, meaning an increase in the population of Argentine stem weevils. Pasture control is going to become harder. This has become more of a challenge. And that's absolutely, nice work, that's absolutely true of all our ecoclimatic systems. I love this little picture. It's a picture of the Southern Alps, and you can see forest in the valleys, you can see alpine meadows higher up, you can see the snowpack. Well, climate change changes all of this. The snowpack goes away, the alpine extends further into the mountains, the forest tries to grow further, and all of this, if you look at the rate of climate change in New Zealand, which is faster than the rate of evolution, our species are unlikely to be able to adapt to these sorts of changes unless they are already pre-adapted to do so. And those pre-adapted species are ones which have broad tolerance and broad range, and that's a pretty damn good description of invasive species. I think that our problems with climate change aren't just the climate is changing and sea level rise. I think we're going to see massive disruption to our ecosystems, both pastoral and conservation, and we need tools to deal with it. So do we have those tools? Well, these are the current tools that we have available to us, sort of available to us. Broad-range pesticides. Well, what we've learned in the past few years, and indeed in the past month, is that broad-range pesticides are an absolute problem. Right? Neonicotinoid insecticides are extremely problematic. Glyphosate, in a paper a couple of weeks ago, has effects on insect melanization, which is its immune system. There's all kinds of problems. So they're going to be removed as a tool. We talk about targeted toxins. Predator Free 2050 is very keen on targeted toxins for rats. Well, I've developed targeted toxins. There's no market because the people who develop pesticides are large international companies who don't give a shit about targeted toxins for us. They care about markets where they can sell broad-range things, one chemical to as many people as possible. So that's a problem. They're also really hard to make, and they likely have off-target effects. We do have tools like gene editing and gene drives, but they're immensely controversial. I don't think we have social license to use them at the moment. Maybe we will get social license. These are useful tools, but currently they're tools that we can't use. Pest trapping and shooting is expensive and ethical implications, and I would challenge you to shoot Argentine stem weevils. And as I've shown you, biocontrol isn't always the golden bullet, right? It involves the bringing in of new species, something we'd like to stop. Oh, come on, can't I have 20 minutes? There are off-target effects, there's failures, there's hitchhikers. This virus that we found, thank God, it really only affects these parasitoids. What if it actually affected our insects more broadly? So what can we do, in my opinion, and what we should be doing? Yeah, go on. Well, I think that the Bioprotection Research Centre and Bioprotection Aotearoa's role here really is in deep research, right? A lot of the time, when we start thinking about our problems, we don't understand them well enough. That's what I've learned about the Argentine stem weevil problem. We've learned a lot. Did we know enough to be able to stop the decline in control? We didn't. We might know enough now, but we don't have the tools. So we need to do the research and understand our environments. We need to understand what's happening in our urban primary production and conservation estate. So for example, in Europe, there's been a 70% decline in flying insects. That came about from a study generated by an amateur entomology society. I am really worried that we are not doing those studies in New Zealand. Have we had a 70% decline in flying insects? Stephen tells me that in the high country, the Mackenzie country, that may actually be the case for some of our native species, right? but we don't actually know because we're not looking. We need to generate the data that supports the use of the deployment of new tools, right? It's really hard to talk to you about gene drives being a potential solution for pests without being able to say, what are the costs, what are the benefits, right? That involves research in the lab to understand those costs and benefits. Nobody will say yes to something controversial unless you can say, this is what's good and this is what's bad about it. I think we need to make a sort of pledge here, and I want you all to raise your hands. No, I think it's really important that everything we do is open, that our work is shared, our results are transparent, it's available to the public and other scientists. Because in the end, the solutions that are going to benefit New Zealand are going to be difficult to implement if they are controlled by individuals who own IP. Let's forget about IP. This is for the good of New Zealand. And I think the most important thing is that we as a group need to provide the people of Aotearoa with the tools, technology, the knowledge that empowers them to build and support resilient ecosystems, right? That's our job. We should be building that knowledge and then providing it transparently to the rest of New Zealand. Whack on the last slide. Acknowledgements. Massive group of people here. There's a lot of people in Project 2, and I just wanted to point out that these people are from the University of Otago, Lincoln University, Ag Research, Plant and Food. There's probably many more in there. So this is a huge collaboration of people across institutions, across New Zealand, and that, I think, is the real strength of a centre of research excellence like this, bringing people together who can actually answer the questions that we like. This work was funded by Bioprotection Research Centre. Some of it was funded by the Ministry of Business, Innovation and Employment, some of it by Genomics Aotearoa, and I am brought to you by the number 7 and University of Otago. Thank you.
Okay, if I could ask the speakers, Jay, Michelle, Peter, come and take a seat and we'll answer questions. And we have a roving mic. Who would like to ask the first question? Come on, you get to play catch if you ask a question. Robin.
Fantastic to hear the word virus, of course. They rule the world. Were you able to correlate that with any impact on it was in the parasitoid or the weevil? It was in the weevil. Parasitoid.
Hello. So it's the parasitoid that carries the virus, and that's not uncommon for parasitoids. Many of them carry viruses which they pass on to their host, and that damages the immune system so that the egg can develop. We don't know if that's the case. We also are trying to look. It doesn't seem to us that there's any decline associated with resistance and that virus, but it would be really interesting if there was. We are now looking. So one of the cool things about this is that Ag Research, when they brought in these weevils, or brought in these wasps, they grew up lines and they froze them. So we're now looking in those lines which were brought into New Zealand to see whether the virus was there. My suspicion is this is a virus that's present in this species and is about allowing that parasitism to occur. But wouldn't it be great if actually we find that it's been derived in New Zealand and maybe curing the virus will restore parasitism rates. But yeah, more research to do.
Can you throw that over here?
Here we go.
Thank you. Could you just go back, Peter, to your second to last slide? Or could somebody? Yeah, that one.
Oh, the randy one.
I think that was it was really good to sort of put it all into context, and I'm wondering if the two others could sort of, I don't know, I guess have a roundtable discussion on what can we do and how can we do it. I do agree that we need to be transparent and open access and all of that. I think there's a bit more to it. I think that there's a bit more to sort of changing the world than presenting our data transparently. And I'm excited to hear more about that this afternoon. But I'm wondering if the three of you and Travis could talk about what can we do and how.
So I had an interesting opinion about what Peter said about IP, forget IP. I slightly disagree. So I think IP has a role to play in the way that we develop technologies and use them. As an owner of IP, you can decide who you pursue for compensation. I think for the greater good, you can decide not to pursue, ideally, the universities or research groups that are trying to do things for the greater good. But if let's say a company came along and decided to deploy it to make money, then you would deploy your IP and say, no thanks, I'll have that money. And so IP has a useful role to play. I think the greater problem is secrecy, and we don't like talking about things that we feel unprotected about. And I think, so this great push in Europe about publishing open source, it's very expensive. That's the problem right now. But then there's a really nice free choice of publishing on bioRxiv. We have options available to us. We just need to normalize them and start utilizing them. And the more we talk about our research with fellow researchers and make it more available, use jargon, well, reduce the use of jargon and open it up to the public, the more engagement we'll have. Scientists have been traditionally very bad at communicating our research. And as a community, there's a huge amount of space for us to change that.
Yeah, what he said.
It's an interesting question, this whole social license, because we went through this discussion in 2000. We had a Royal Commission a year long, came to the conclusion, proceed with caution, which we didn't. And I don't see that much that's actually changed in the general society attitudes, despite more education around what genetic techniques are. So I don't really see how we're going to go forward with some of these tools in the current environment, and I don't see how that environment's going to change. So I'm a bit negative about it. So.
I'm not sure what my opinion on this is. I do think there's definitely a need to share more science knowledge with decision makers and people who influence decisions so we can make more informed decisions. I think when it comes to the question of social license, I actually agree with you, Travis, there, because I think the problem with the way that many of us make decisions, we sort of, we kind of plan from meal to meal quite often, for the most part, very short-term thinking. And when we're outside of, sometimes when we're in our work, we can plan for longer than that. But, that is such a difficult, difficult kind of, I guess it's just the way that the human condition really, it's such a difficult place to bridge those two sort of styles of thinking. So I don't have the answer.
Embarrass myself, that's good.
Thank you. Thank you very much. Oh, that is working. Thanks very much for those talks. And to this issue on open science and sharing results and making more freely available, also for the public maybe to decide. It's also quite a tricky space to be as well, because a lot of people have their own biases and own opinions. And I mean, even if you are sharing 20, 30 papers saying this research is good, people find one paper that says, no, it's bad, and you can base everything on that. I suppose it's also educating about the scientific method and how that works, and how scientists work, and how we actually make these decisions. Yes, you are going to get negative results, things that go against it, but there's other work that can say it's a good avenue. I suppose making people aware of that is also important. So yes, sharing information is good, but also teaching people how to use that information responsibly is also good.
I agree with you, and I think that this is the way that we, as scientists, have approached the kind of generation of new technology. But I'm intrigued by the fact that I think the thing that has changed the world most in my lifetime, the smartphone, it's changed huge amounts of things in good ways. I can find my way back to the airport, and in really bad ways. Facebook owns everything I do. There's been no discussion, right? Nobody ever came out and said, here is a new technology, this is what it may or may not be able to do, let's have a discussion about it. So I'm intrigued why, I'm not saying that we don't need those discussions, in fact perhaps that we did need to have those discussions, but it's interesting to me that some things, like physics for example, we've thought of it, we've implemented it. Biology, we've thought of it, we can't implement it. We won't implement it. You know, genetic modification has been around for a very long time, and there are not massive indications that it is problematic as a technology. You know, the use of it is always, and the same with the mobile phone, you know, good uses and bad uses. So it just intrigues me and then we take the view it's about, you know, that scientific process and about teaching people about the scientific method. I don't think it is. I honestly think it's about benefit, that we don't talk enough about the benefit and we don't demonstrate that benefit. When the Royal Commission was there, what everyone talked about were Roundup-ready crops, which the benefit goes to Monsanto. Well, they're rich enough, and they're not very nice people. Right? But what if it's the benefit is to us in the control of wilding pines, or the benefit is to us to get rid of wasps out of our native forests? Then I think we can have a discussion. But you've got to know the risks and benefits, because without that information, we're lost. And so we need to be able to do the research in containment to test those risks and benefits. That I think is the information we're lacking. When we put forward scientific information, we say, blah. But actually if you can say, this is the benefit to you, this is the risk, then it's the same decision as crossing the road.
Sorry, I just wanted to add a little bit to that. Obviously going to come off as really naive, but we've seen this before. So when, going back a little bit, so that when electricity was first developed, there was the AC versus DC argument. And so they had all this propaganda from the DC Thomas Edison group where they went around showing people that you can get electrocuted on AC currents, and everyone was against it. But when the obvious advantages of AC current came out, everyone switched. It wasn't instantaneous, it wasn't rapid, but it did happen. So, I mean, historically, we inform ourselves from these lessons that we learn. And so I am naive enough to believe that change is still possible. It's always possible until we give up.
Let's give up.
Oh, I wish you'd all sit together.
I think it must be your turn.
Right, thanks for that. As somebody who actually does research into decision making, I'd like to make a few comments. One of the things we know from a long line of research is something you've touched on, which is the one or two economists in this room would know about, which is delay discounting. We know that people aren't going to act unless it's pretty much in their face. Okay, so, and that's one of the reasons our response to the other pandemic, Peter, the COVID one, worked so well. It was immediate, and it was highly threatening. What that means is that we actually need to find out what people value. And I'm glad to hear the word value come up a couple of times here. And there hasn't been, Travis, there hasn't been a lot of research done that way in this area, which is one of the reasons we're on board. But I just wanted to say thanks for pointing that out. It's nice to hear social science and behavioral science being brought up as an important issue here. So thanks for that.
You're off the team. Questions? Stephen?
Can I just help Dearden with some of the things he said about me? I'm sorry. He gave the impression that I collected one parasitoid off one lawn and brought it back. I do need to point out we brought in 140 clonal lines, which we couldn't identify as clones because we didn't have the technology to differentiate between them. We can now, and so what we did was released equal numbers of everything all over New Zealand to see what did best where, and I think we can do that now. Now the important thing about that is we can look at traits to see what traits there are that predispose to success. And this is where I think Peter's points come through really clearly. Once we know the traits, then we can select for those traits or manipulate for those traits, and then we can take a lot of the uncertainty out of biocontrol, which still is based on some pretty broad brush reasons for selecting one agent or another. So I think this is where the ecology and the genetic range intersects with a lot of the work that Peter was talking about. So I think that's what he meant. Thank you, everyone.
Okay, I've got a question primarily for Michelle. In the Bioprotection Research Centre, we focus very much in trying to help productive industries, our primary industries, by being more sustainable, providing them with solutions. And I think we've had some success at doing that, and I think industry appreciates the research that we've done. But your talk was very much about consumer perspectives, and I wondered to what extent the research that we do translates to benefits to consumers, in that the savings that industry makes through the science may not necessarily get translated to the consumer. And primarily I think that I can buy vegetables and fruit at the local green grocers much cheaper than I can at New World. And there's clearly a big markup on fruit and vegetables by industry. And does that mean that the benefits from science don't ever translate to cheaper fruit and vegetables? I mean, is there a problem there that no matter what science we do, those benefits don't get translated directly to the consumer?
Yeah, I mean, I think you raise an interesting point there in terms of our, the way that, in terms of the domestic market, yes, I mean, the value is being captured along the way, like not necessarily by the grower, and sometimes it's by the supermarket, and often it's by the marketing and branding. And so one of the things that we see is that people are willing to pay not so much for the product, but for the experience, or like put a cool name on it or a label. That's the part that people will pay for, and perhaps that's to the that weird thing about needing to understand that immediate value in that moment. So but the other thing that I was talking about was really about how we can integrate more values into our decision making, so we can start to think about how we allocate resources and to create more benefit for our wider society, as well as through to businesses. So, but yeah, I'm not too sure beyond that.
Further questions? I feel like I need to bounce it.
You do need it, right? I've been told I need it. Jay, your wonderful work in PSA, was that done largely by you and your team here, or what other groups were approaching it? And if so, how much collaboration and how much competition to get there first?
Right. Thank you for that question. Sorry, I suppose I did the larger group that I work with a disservice. So the mutations I did myself. I have to thank Matt Templeton for the opportunity to, first of all, give this talk, but also the opportunity, because he's the one who applied for the postdoc funding and got it, and I just sort of jumped in and started doing the work. So the vector system, the knockout systems, that was developed technology. The knowledge that we were basing all the work on, that was all through publications that we had access to. The knockouts themselves, day-to-day stuff, was all done in-house. I did it, but also in partnership with some students that passed through the lab, some honors and master students. But yeah, science is never really done on your own. I can't think like right now of all the people that contributed to it, but there was a lot of contribution. I did not do it all on my own. Thank you for the opportunity to address that. Yeah.
So there's a follow-up, which is a statement, not a question. Am I allowed to do that?
Yep.
So the statement, I don't know whether this is going. So the statement or observation is the power of collaboration and the ability to make the boat go faster when everybody's putting wind into the same sail. And I guess with the various hats I wear around the science system, the secrecy and protection of some work and the out-and-out competition, which slows the boat down. And I would like to encourage thinking around how, and I think as has been articulated today, how the mechanisms for amplifying effort to solve these problems quickly is a far greater goal than individual success. So maybe I can just share that and reflect on it over the next couple of days.
That moved a long way. Firstly, I'd like to thank all of the speakers and just a comment about the IP, particularly at the for bioprotection, the work that's done here. That IP is at a very high level in terms of blue sky science. It should be, I agree, Peter, it should be openly shared so that the more commercial ends can be developed faster. But my question really to the panel is about, Peter, you put up a slide talking about the number of species, indigenous species that were under threat, and the implication was that that threat is accelerating because of climate change, amongst other things. And yet we seem to be in a situation where our ability to make decisions is getting slower. So we've got a rapidly accelerating threat and a reducing ability to respond to that. I'd really appreciate any comments the panel might have about how we can accelerate decision making in this country, and some of the questions are actually up there on the screen about gene drive, gene editing, those sorts of things, but a whole bunch of other things, resource management decisions, land use, a whole raft of things. How do we accelerate our ability to make decisions and actually implement change?
I agree that we have a problem in terms of the time that it is taking to make decision, and in resource management, you know, we have processes that decisions, you know, plans that are supposed to last for 10 years, that take 10 years to be sort of agreed upon. And so, you know, growers operate within an area of uncertainty which limits their ability to invest and actually limits our ability to achieve outcomes because of the uncertainty. How we get over that, you know, I'm not really sure what the solution is, but I do sometimes think we have got to a place where we've overcomplicated the process. We allow a sort of an endless amount of appeals on detail and for decisions to be relitigated over again. But on the other hand, you know, I know in our work, we see issues where, you know, in horticulture, I kind of alluded to it in my presentation earlier, where a lot of our resource management is kind of for the dominant activities in New Zealand. And so when you're a smaller sector, you know, sometimes the rules simply do not make sense. And so you have to go back and challenge them. So it's actually really, you know, it's not simple. So I guess on the one hand, I would say perhaps we could make decisions more rapidly and perhaps for a shorter period, you know, review them, you know, more frequently, but to create certainty in sort of blocks of time. But of course, some decisions are sort of irreversible as well. So I understand the problem. I'm not sure what the solution is. Maybe you know.
No, I can't solve your problem, but I do think. No, I'm out. I think that one of the things I've been quite impressed by is the ability to get information to decision makers faster. Right, so I think that the pandemic has been a really quite salutary lesson in the standing capacity of biologists in particular to provide information and to do that quickly and in a way that can be dealt with. Now that's probably to do with the immediacy that our friend at the back, whose name I didn't catch, pointed out. But you know, the genome of COVID-19 was sequenced in 24 hours by a group in Australia. You know, we put in place things for sequencing the COVID virus. Those sequences take well, I don't know why they take 24 hours because I could do it in 8. But you know, there's information there that allows for rapid decision making on a small scale. But that small scale decision making is things like shutting down Auckland or presumably, you know, shutting down Wellington in the next day or so. So the fact is that some of biological science has sped to the point where actually we can get very detailed information to base decision making on quite rapidly. That doesn't help your process problem, which is, you know, is the process to make the decision too slow? But I do think that as scientists, we have some kind of ability to really get the data on which that is based relatively rapidly. You just need the immediacy, the need for that information. I think we need to spend a lot more time talking about what the consequences of some of the problems coming down the line to us actually are, because I think, to be honest, I think New Zealand is complacent about climate change, and I think it's going to smack us in the face a lot. That didn't answer your question either.
Now, kia ora tātou. On the last point about providing people with empowering knowledge, I'm involved in a program with our Land and Water that's trying to address this problem. A small group of scientists, a large number of enterprises working more or less in perfect competition with no spare resources. We commissioned a report from KPMG who said that industry organisations like Hort NZ are an obvious vehicle for amplifying impact of science. So just while you're there on the panel, I'm just wondering if you've got any thoughts about how an organisation like Hort NZ might take best science and amplify it in a way where members would be able to access it more easily than resource-constrained small enterprises otherwise can do.
I think that is, I do think that that is a good idea because we have, we have access to growers and we produce information that they read and we see them in a range of contexts. And one of the things that always sort of interests me about growers, my area is in environmental science, but when I go and speak to them, of course, they are sort of have expertise across such a range of disciplines. So I think it is a good idea, and it's not one that we really do that much at the moment. We tend to speak, quite specifically around narrow matters or at quite a high level. So I think there's absolutely an opportunity to improve that communication through that vehicle.
Yeah, I work quite closely with Apiculture New Zealand, and I think they have a science industry group who discusses these things. Tomorrow I have to nip out of here and present to their conference about some work, and so I do think some of these bodies are doing really well, and I also shout out to the CRIs. I think they're doing a really good job of getting in front of those bodies and saying, what things do you need in terms of science, and how can we help you transmit that through to change growers' performance, not performance, behaviour. Make them do different things. There you go. Sorry, I've only got a certain number of words, and I think I ran out.
Okay, if you'll join with me and thank our speakers for an excellent question and answer session as well. Okay, two tasks before we go to lunch. First one, apparently the places in Wellington that are of interest are now online, so if you're from Wellington, perhaps you should have a look. And before you get to eat, we're having a photo taken out front of the library, so the faster you get to the front of the library and get your photo taken all together, the faster we eat. And then we're back here at 1:30 for the next session. So thank you all. Just before we start the session, just an update on the COVID situation in Wellington. My understanding is that Wellington has gone to level 2, that people from Wellington should take their level with them. So the people who have come down from Wellington, the advice is that you are at level 2. That includes not being in meetings of more than 100 people. I don't want to count, but we'd be close.
It's roughly around 100
Yeah,
or not
rough.
less.
So 97, thank you. We have spoken to the people from Wellington, and my understanding is that the majority of them didn't leave their houses on the weekend anyway, so because of the rain and work. One person was here this morning who was in a place of interest and has been off talking to Helpline this morning. So at the moment, with no further advice, we will continue. I'm quite, if you do not feel comfortable with that, please make your decisions. And I'm trying to get advice about the dinner tonight as well. Any questions on that, or?
It'd be good to know if there's people not going to go to the dinner.
Yeah. If at this stage you don't think you'll go to the dinner and you had signed up, please let us know, because we'll have to let the caterers know as well. If you have decided not to go to the dinner already and you were signed up, please let us know, let Sandy know, so we can let the caterers know. All right, if there is, and we will be seeking further advice during the afternoon. Juliet is here somewhere, and I suspect she's busy on the phone at the moment, but as soon as I can get hold of her, I'll find out what the latest advice is. Okay? Thanks, Eddie. Don't pinch my things. Right, I would like to invite Professor Paul Dizelle up to chair the next session. Thanks, Paul.
Thank you very much. Kia ora tātou, haere mai koutou ki tēnei hono anō. Kei te mihi ahau ki ngā ringawera mō o rātou manaakitanga ki a mātou I tēnei rā. Ka nui te mahi a mua, nō reira mera whina tātou ki a tātou I tēnei kaupapa. Tēnā koutou, tēnā koutou. Kia ora tātou katoa. So I've been pleased to acknowledge the cooks and the people who have looked after us at lunchtime. There was a real buzz out there, and it's an important part of a gathering like this that we are well looked after. So a big thank you to them and all of our hosts, and to say that there's a lot of work in front of us, and so let us help each other in this work. This session, before and after afternoon tea, is about science-led policy, and we have a distinguished panel of people who are going to share from their knowledge about science-led policy, beginning with Professor Anne-Marie Brady. Professor Anne-Marie Brady is a political scientist. Her groundbreaking research into CCP political interference activities has demonstrated the important role of the academic as critic and conscience of society. Professor Brady is a specialist of Chinese politics, both domestic policy and foreign policy, polar politics, Pacific politics, and New Zealand foreign policy. She is a fluent Mandarin Chinese speaker. She is a founding and executive editor of the Polar Journal, and she has published 10 books and more than 50 scholarly papers. She is a global fellow at the Woodrow Wilson Center in Washington. In 2014, she served a 2-year term on the World Economic Forum's Global Action Council on the Arctic. Her research has a strong policy focus. Anne-Marie, I acknowledge your mana and knowledge on this kaupapa, so please come and address the group.
Tēnā koutou katoa. Warm greetings, everybody. It's a great honour to be here, and I do feel a bit fish out of water, being a political scientist speaking in a room of mostly scientists. But I think what connects us Well, there's a lot that connects us actually, but one of the things that connects us is that many of us are interested in doing work that is useful to our society. And we hear the word policy a lot, and it's the political scientists who theorise the policy, or ideas about policy and public administration. So I want to share with you some of my observations about the state of academic freedom in New Zealand, something that concerns us all very much, and also what's called the critic and conscience role of the academic. It's written into the guiding legal document of our New Zealand universities, but I would wonder how many of you have read the Education and Training Act. Could you put your hands up if you have? Oh, bravo. There's 3 or 4 of you. The rest of you, please go have a look at it. You ought to, because the universities base their policies on it, or they're supposed to. Well, according to, there's many things I'll explain to you in a minute about the Education and Training Act of 2020. It was updated. The original one was 1989, and I think they're our forebears who wrote it, because not every society in the world has a piece of legislation like that, and we are very, very fortunate. It's very precious to have some of the rights that have been enshrined in that legislation. Our academics have a crucial role in a modern democracy. We can offer informed commentary, independent research, and provide new ways of thinking. And yet New Zealand scientists and social scientists, as has been demonstrated in academic papers, and as I have experienced myself, have been under increasing pressure. It could be from corporations or even foreign governments, to avoid raising uncomfortable truths. And our New Zealand universities are dependent on external funding to balance their books. They don't champion or promote academic freedom. And some universities actually appear to be putting limits on academic freedom and pressuring their academics not to rock the boat. So I'm going to tell you a little bit about your rights and, in fact, your role within the legislation to perform the part of the critic and conscience of our society. And I'm going to talk about a little bit about my own experience of being a critic and conscience, and show you how you also could make a contribution to do that. And I include the practical aspect because I know myself, when I was thinking about, when I realised that I'd had some information that I felt that the New Zealand public really ought to know about, I didn't know initially how I could make a contribution to our society. And I have learnt since many, many ways that I think more of us ought to be able to do as well. And they're not that hard. So first of all, let me point your attention to the wording of the Education and Training Act. And the crucial words that I'm looking at is the highlighted ones, that universities accept a role as critic and conscience of society. That is what they are supposed to be doing. It goes on to say about academic freedom, the freedom of academic staff and students within the law, that universities should be able to have this, that they would be within the law to question and test received wisdom, to put forward new ideas, and to state controversial or unpopular opinions. So that's what our universities are supposed to be encouraging us to do. And on university leadership, particularly our vice-chancellors, this is what they are supposed to be doing. They should be imbuing the employees of the university with a spirit of service, both to the institution and to the wider community, in other words, our democracy. And they should be managing the universities in a responsible manner that reflects the needs of academic freedom. But do they? I had a look at our collective employment agreement at the University of Canterbury, and in our collective employment agreement it says right in the first section on the terms of employment, that staff shall have the right within the law to act as a critic and conscience of society. I also read the employment agreement of the Lincoln University, and I found that you don't have that wording there. So for those of you at other universities of New Zealand, you might want to look at the wording of your employment agreement. Now, just because it's not in the employment agreement doesn't mean that the Employment Act and the Education and Training Act doesn't apply to you. But it is good to have it in the collective agreement. So those of you active in TEU on your campuses, I reckon that you might want to move in your next bargaining to have that inserted in there, because I think we do really need to remind our university leadership these days that we have a specific role in our society. Our government and our people expect us to be the conscience and critic of our society and expect that we are enjoying the benefits of academic freedom. Now, our Education and Training Act and the policy that is at my university on academic freedom is based on international thinking on academic freedom. So the 1997 UNESCO recommendation concerning the status of higher education and teaching personnel stated that academics should have freedom in carrying out research and disseminating and publishing the results thereof, freedom to express freely their opinion about the institution or system in which they work, freedom from institutional censorship. So it's not just New Zealand, but it is an international norm that we academics will be playing the critic and conscience role within our societies, and that our societies will support us in that. However, we are in a rather dire era, as you might have noticed, the era of misinformation, of disinformation, and unfortunately we also have a weak media. And in political science we call the media the fourth estate. They're a balance in the check on power. But our journalists don't have time to do in-depth reports anymore, or even read anything in-depth that we might have written as academic specialists. So in this time of a weakened information environment where there is circulating harmful ideas, we have some very big challenges facing our societies. The global political order is shifting. We are seeing breaking of international norms, and New Zealand's security is very reliant on a rules-based international order and the level playing field. We're seeing great environmental challenges such as climate change, and of course we're experiencing a pandemic, the like of which we have never seen before. And we need all the more than ever before that expertise from the academics, the critics, and conscience of society who will raise those sometimes uncomfortable truths and provide information to enable our legislations to pass good new legislation, and for our society to understand the reasons why. And David Edwards from UNESCO has spoken on this in the context of our current international environment. And he says that a free teaching position must inculcate students with the values of democracy and the competencies of healthy skepticism, critical thinking, scientific methods, understanding of history and media and internet literacy. So I want to share with you my own experiences of stepping up and being a critic and conscience in our society, and also internationally too. I was your typical bookish academic, a nerd, as one of my friends calls me. And it was material information that I found in my own research and in conversations that I had with foreign policy practitioners within our government trying to share the information that I had, and thinking, initially, well, our government will have this all under control, won't they? They'll have the answers to this, and discovering that that wasn't the case, that I began the journey of having a increasingly public role. And I can tell you it was very uncomfortable. I am not naturally an extrovert. I like people, but this public role is not within my normal comfort zone. But I stepped up because in the end I knew that no one else would, and that I had knowledge and expertise that would be useful for our government and for many other governments. So the first thing that I got involved with is through my research on China's interests in the Arctic and Antarctic and their wider maritime strategy, I began to wonder how our lovely country, Aotearoa, New Zealand, could face up to and meet the challenges of the changing global order. And it wasn't a simple answer. It wasn't just about our one bilateral relationship. We had to look at a whole series of our relationships, of our international partnerships, and also our involvement in international organisations. As I said, the global order is changing, and the international norms are threatened. So our participation, both in the international system and our relationships with individual countries, needed a new proactive strategy. And so eventually, seeing that no one else was going to do it, the government doesn't have the resources for this kind of in-depth thinking, I gathered together a team of academics, first international partners, specialists on small states theory, because New Zealand is a small state, and then gathered the New Zealand academics for focusing on the New Zealand aspect of this for a project I called Small States in the New Security Environment. And we called it a preparedness initiative, so proactively looking ahead of what New Zealand should do. The work was meant to be, and is, policy relevant, and yet theoretically informed. And I made my participants write 1,500-word policy briefs. We later turned them into chapters, so we could tick the box for our universities and have Scopus outputs. And they're more in depth, but even the writing of that edited book is, I told my authors to write like you're writing for the New York Times, and short sentences, active voice, and so on. And so these 1,500-word policy briefs are the sort of thing that a minister can read, as I would say, but in the limousine from the airport to their next destination, and focused on very specific policy issues that we need to think about more. And the policy briefs, the audience that we imagined was for the politicians, but also for the media, and there have indeed been stories that have been built off our policy briefs. I also organised 2 events. The first in 2017 was a hui on New Zealand foreign policy, and I called it a hui because the format was not talking at, but engaging, and it was a safe space where we exchanged ideas on very sensitive topics, and all the more so for the 2018 wānanga, which I organised at Rapaki Marae on New Zealand-China relations, where we had senior politicians, policy people, as well as academics attending and sharing our experience. I also set up a pop-up think tank because I didn't want to waste money on overheads. And so it's still there. You can have a look on it on our website at the University of Canterbury, the SANS pop-up think tank. Last year, I set up a special series for doing some forward thinking about what we're going to do in this post-COVID-19 scenario in our foreign policy and our economy and so on. So that was my first foray into doing foreign policy work and stepping up into this critic and conscience role. And the next actually began about the same year, just by chance, was my research, which raised the issue of the Chinese Communist Party's political interference in New Zealand, but also internationally. And it was just one of those things. I was invited to be in an edited book project, but what I found in my research was well above and beyond anything I'd wait for 3 years of academic publishing to come out. and it was indeed 3 years before the editorial book was finally published. So I released my conference paper ahead of time. It's called Magic Weapons, China's Political Influence Activities under Xi Jinping, to the think tank that I'm affiliated with as a global fellow, because they have the platform there, and it was immediately got international attention. I was interviewed by New York Times, and I don't know how many interviews I did in the next 2 days after that, and had a huge impact on strategic thinking in the US. It was earlier this year acknowledged by the National Security Council of the US that it helped to shape the Indo-Pacific strategy, and has had an impact on international thinking of many countries, from the UK to France, Denmark, the Baltics, and so on. And it's 57 pages long, and it has hundreds of footnotes, because I wasn't going to be talking about stuff like that without a lot of evidence. But I knew that many politicians, as well as journalists and other interested people, would be afraid to read a document as long as that. So I wrote a 1,500-word policy brief in 2017, just as the coalition government was announced. I released my policy brief, which summarised a new template for New Zealand-China relations. And to my delight, my words have been echoed by our Prime Minister and our Foreign Minister this year about strategies we should follow. And I know I've been confirmed by officials that they have, in fact, very much, that my work has really shaped a lot of the things that have happened. Although you wouldn't know it from the media reporting on it, which continues to be very weak on this issue. As I mentioned, I organised a wānanga in 2018 that helped to move the debate a bit forward, that was already underway internally within our government, whether we would balance, which was more important, national security or economic security. In the end, thankfully, national security won out. And then also in 2018 was when the government decided to launch an inquiry into China's political interference activities. And at first it was meant to be closed door, but I heard about it by chance and was encouraged to apply, and which I did. And then there was a media storm in February about it, and I encouraged more people also to write, and so that they wanted to give testimony. And so it was just this natural, organic thing where our society was able to participate in the wider society in the policy process. And then there was a further parliamentary inquiry last year into political interference in local government. And it's led to new legislation on political donations, the reform of the Overseas Investment Act, changed national security priority. Foreign interference is the top priority of SIS now since December 2018. There's going to be change in local government. Also, I draw your attention to this trusted research guidelines with which the Universities New Zealand and SIS have been in discussion with for a while, and it was very quietly announced in March this year. All of us individually would be responsible if we had an unfortunate relationship with where you're passing on military sensitive or police-related research or information, know-how to a partner university in China that was connected with the PLA, or even if it wasn't, in fact, according to the new legislation that was, or changes to the management of this kind of research in New Zealand. So you can have a look at the protective security link there. If you haven't heard about it, you should look it up. And you can look at the change guidelines on the strategic goods, which was updated in October last year. So what I have done is I've done lots of parliamentary submissions. I've given oral testimony to Parliament. I've done 20 op-eds, 9 policy briefs, met with lots of New Zealand MPs, given presentations in parliaments of Australia and Canada, and talked to many, many governments, plus lots of media commentary. And I created a basic template for how to do a policy brief, which you are welcome to borrow from. And you'll see on my pop-up think tank that all the policy papers follow that guideline. and my op-ed as well. I taught myself how to write an op-ed, and it was confirmed to me by one of the top, the Asia editor of the Financial Times, that I have it right. You begin with a news peg, you have to have a strong opening sentence, and the journalists call this your ball statement. We'd call it an ovary statement or a lead. What is it you're trying to tell me? What's your opinion on the news? And don't make it any later than the third paragraph. You must have short sentences. You must use active voice. Persuade me. Make me want to read this thing. And end it with a grenade in the last line, if at all possible. And for the parliamentary submission, I didn't know how to write a parliamentary submission when I started. You might want to tell people why you're the person to comment on this issue. You need to respond directly to the terms of the inquiry. To my amazement, I found others participating in the inquiries I was involved in did not reply to what was actually asked, went off on their own tangents. Well, that's not helpful. Provide some expert policy advice. Try and connect what you're saying to the existing legislation. You can help the MPs to improve on that legislation. And again, active voice, avoiding jargon, short sentences. And when it's your chance to give oral testimony, speak to the MPs, give them something new, and be prepared to be challenged. I wasn't prepared the first time round. They set someone up to disagree with me, who, without telling me, who wasn't even on the panel. But that's part of the process. You've got to be able to argue your point. And then afterwards, you're going to have to talk to the media, and you might want to write an op-ed. So the concluding paragraph of my famous paper called Magic Weapons went as follows. Democracies have magic weapons too. We have the right to choose our government, balances and checks on power through the courts. Our regulatory bodies, such as the Commerce Commission and the Press Council, the legally supported critic and conscience role of the academic, freedom of speech and association, and the fourth estate. Now is the time to use them. Thank you.
Thank you very much, Anne-Marie. Anne-Marie's got 1 million papers to mark back at the University of Canterbury, and so she has agreed to answer some questions and answers now for about 10 minutes, and then we will release her back into her office. So we're just doing the magic ball. People know the trick here. Just being switched on. Here it is. I've seen this hand first. I know it's at the back. But
Thanks very much for a fascinating talk.
Can you identify yourself?
Yes? Batman. Robin McDiarmid from Plant and Food Research, the University of Auckland, associated with the Bioprotection Aotearoa, President of Australasian Plant Pathology Society. The reason I say all of those things is I'd like your comment on how you negotiate multiple hats and taking this critic forward. Yes, when you've got all of these hats, and some of the organisations or societies will be in different situations in terms of that freedom. If you could lend some advice, that would be really gratefully received.
Okay, thank you very much for your question. I don't have any doubts about my different hats. I just think of myself as an academic, and my contract with the university and the most important legislation to do with me in my role tells me that I have the critic and conscience role. So I act on that basis, and so far it has kept me well. And so I don't worry about any of the other associations. I don't think that they are in conflict at all, but no, I don't. I just imagine myself as the academic who's been paid, have the luxury of being paid to be the critic and conscience of society. And that's what the legislation tells me as well.
Great, thank you very much for a very useful and interesting talk. You touched on the issue. There seems to be some quite significant shifts in politics globally, and you look at the UK and the States recently. Have you got any feeling or thoughts on what seems to be driving quite a global shift in the political environment? And that's outside kind of China and the communist countries. I'm talking about the democracies.
Well, I mean, there's different things happening in every different society. You know, you've got you had Brexit in the UK, you had the coming to power of Trump in the United States, but I mean, Brexit had been developing for a long time in the UK, and the polarisation and of politics in the United States happened for a long time too. Unfortunately, I say unfortunately, humans are rather stupid and we don't learn from history. And so what I see, I've been saying for a long time, and I didn't want to be right, but fortunately I think I am, that we're in a long 1930s. So we've seen a lot of this behaviour before the rise of nationalism, the splitting away, a tendency towards appeasement when you see an aggressive power. And we know what we need to do, and it's just rather uncomfortable. But we've already made some big changes last year with COVID when we were faced by this challenge. So I think, and you can see that our government also understands that challenge, and it's part of their internal planning. And what I keep railing against actually is our Minister of National Security, that she never speaks on the topic that is our Prime Minister. She's also Minister of National Security. So I think that what's going on now is we have seen it before in human history, and now, but the kind of warfare is different. It's not kinetic warfare, it's hybrid warfare. It's very important that the public understand this, as well as the public sector. When I was first starting my research that led to the Magic Weapons paper, I interviewed one of our former senior politicians who about 10 years before had raised similar issues to me in New Zealand Parliament, and I asked him why he had stopped raising those issues. And he said that was because the media and the public didn't understand what he was talking about, and they thought it was just sour grapes. And he said without the public conversation, you couldn't get the policy change. So what I've learnt to understand is like there are two wheels. So that's why I want to talk to you today too, because the government can only do so much if the public wheel is not moving as well. And that's the same on all the issues that we care about, whether it's climate change or epidemics or geopolitics.
Time for one more question.
Thanks. A quick one. Your start to your address was about academic freedom, and the two points of responsibility. One is a conscience for society, and at the same time protecting the establishment from which the academic comes from. And I'm not quite sure how you reconcile those two, because they can be in conflict from time to time.
Well, that's on the Education and Training Act, but we are also guided by every other legislation in New Zealand, and there's the employment legislation where you have to act in good faith. So I think that's a normal, relationships should always be in good faith. But if you look at the documents of all our universities, their founding piece of legislation is the Education and Training Act. So that means it's above everything else, and we are authorised to critique our own institution if necessary.
Well, thank you very much, Anne-Marie, and because you are a guest, we have a gift for our guests, but not for the members of the centre, sadly. So I appreciate that you are very busy at this time of the year, and you have given up your time to prepare and present to us. So I ask you to join me in thanking Anne-Marie for her contribution.
Thank you. Thank you very much.
No worries. Okay,
Sorry, just before you do, I'll just, latest update for you is the advice is that we are okay to continue. We do have face masks now out in the foyer if people would like to wear them. That's your choice, and we will go ahead with the dinner. So, okay, thanks everyone.
Thank you very much.
Oh, and please introduce yourself when you're asking questions so people online know who's asking.
Very good. Any questions for Travis? No? All clear? It's my great pleasure now to introduce Dr. Melanie Mark Shadbolt, who is a former member of the Bioprotection Research Centre. She was the Māori Research Manager and kai arahi for the centre and did some very distinguished work while with the centre. She is now the Kai Hautu Chief Māori Advisor for the Ministry for the Environment, and she is also the Kai Whakahaere, the Chief Executive and founding member of Te Aotearoa Whakamataki, which is a charitable foundation aimed to provide Mataronga Māori solutions in the biodiversity sector, with a particular focus on ensuring protection of Aotearoa's biosecurity systems. So she is particularly well qualified to speak to us on science-led policy, being a scientist of good, outstanding reputation herself and now serving in the public sector. So kei te mihi ki tau mana me o tau mōhio ki tēnei kaupapa, and welcome Melanie to speak to this gathering now. Thank you very much.
I have to have a wee laugh, don't I, when I hear that. Kia ora koutou, ngā mihi nui kia koutou. Ko tararua ngā pai maunga, ko Wairarapa Moana ngā wai, ko rua mahanga te awa, ko Takitemu te waka, ko Kahanunu te tangata, ko Ngāti Kahanunu ki Wairarapa te hapu, ko Papawai te marae, ko Ngāti Kahanunu rātou ko Ngāti Prau, ko Te Aroa, ko Te Aotearoa, ko Tūwhari Toa, ngā iwi hoki, ko Melanie Mark-Shapot tōku ingoa. I'm Mel, I know many of you, and it's like coming back to a family reunion, so it's been quite nice. I've enjoyed hanging out with you all. I am, as you said, the CE and one of the founding members of Te Tira Whakamātaki, so I have to acknowledge the other two that are in the room, Amanda and Nick, who is hiding somewhere up there. So kia ora to you two. I'm also the Kai Hautu Ngātahi, or the co-director Māori of the Biological Heritage National Science Challenge, so ngā mihi to one of my bosses who's sitting here as well. And I am the freshly minted, so I have changed roles at the Ministry for the Environment. I'm now the freshly minted DepSec, or Deputy Secretary Tūmata Kōkiri Māori Rights and Interests. So a number of hats. I only have 8 days left with the challenge, and then I can free up one of my many roles. I just want to acknowledge members of the Bioprotection Board who are here. Kia ora kōrua. I want to acknowledge the kāhui who are here as well, but also now board co-chair, so kia ora matua. Lovely to see you again. Travis, my former boss, and many of my whānau. It is great to see you all. Genuinely is. I had a wee chuckle when you introduced me, because people always make me sound like I actually do good stuff when I just feel like I lounge around and have free lunches and hang out with my mates. So I've been asked to speak today about a tiriti-led science policy, ensuring our Māori research informs decision making. I've actually been asked to speak about this topic a couple of times this year, and I end up just kind of winging it and going off on a bit of a tangent. So we'll kind of see what happens today. Apologies for those of you who may have already heard me at B3, probably you have, and at the Predator Free Summit, because I'll probably cover the same stuff and or get on the same kind of tangent. But I'll try and do some other things as well. I wanted to take you, by answering this question, I wanted to take you through a really fast journey of what I think a Te Tiriti-led science looks like, what public policy is in my mind, what that science policy interface looks like, what a Te Ao Māori policy interface looks like, and then how we move towards Tiriti-led science policy. But I probably will get distracted, as I said, and end up talking about some of my observations I've made over the last 2.5 years in the Ministry for the Environment. The nexus between science and policy making has a significant impact on how the Crown determines the future of Aotearoa. Public policy sets the rules by which the country functions, while science advice provides evidence and helps to set the direction. Currently, the mainstream view of the science policy interface values objectivity and universality. It draws sharp boundaries between knowledge and action and situates experts outside of communities. This is in stark contrast to a Te Ao Māori approach, which sees knowledge and action as intertwined, is open to different forms of knowledge and expertise, and values tikanga as a guide to ethical behaviour. This means Māori have limited opportunities right now to influence the science policy interface. And this has been particularly evident during COVID-19, when Māori knowledge was often excluded from decision-making tables. Accordingly, a major rethink of the science policy interface is needed to reflect Te Ao Māori perspectives, aspirations and priorities, as well as our rights, our obligations and the promises that were made under Te Tiriti o Waitangi. So from the outset, it's probably worth clarifying what I think a Tiriti-led science system or science is. In my mind, Tiriti-led science is the acquisition of knowledge underpinned by kaupapa Māori methodologies, which are strengthened by tikanga and mātauranga Māori, and importantly, Te Tiriti. It also means that the science that we conduct responds to and understands the promise made in Te Tiriti, a promise to protect taonga and ensure the chiefs and sub-tribes, i.e. our hapu of Aotearoa, New Zealand, have unqualified exercise of their rangatiratanga over their lands, their villages and their taonga. Why do I focus on Te Tiriti or Waitangi, not the Treaty of Waitangi? Firstly, because it legitimises the presence, our presence here in Aotearoa, New Zealand, and governance by the British Crown. It is the original tenancy agreement between Māori and Tangata Tiriti, and therefore it must lie at the heart of ethical science in this country. The Reo Māori version specifically recognises, and is often ignored, the reality that all of the hapu and chiefs' discussions around the treaty in 1840 were in Te Reo Māori. That's not surprising, given it was the language of the land at this time. And unsurprisingly, it was the language that was used to conduct discussions around the proposed treaty. It's also the version that our rangatira signed, about 500 of them, apart from a few rangatira at the heads of Waikato River who were shown only in English text and signed it, so that's about 40 of them. It is the version recognised by international law and the one given extra weighting by the Waitangi Tribunal. It is important then that as scientists and researchers in Aotearoa, New Zealand, receiving funding from taxpayers, the Crown, we are able to apply the articles of Te Tiriti to our practice, in part as a pathway to countering systemic racism and advocating decolonisation and anti-racism. In Te Tiriti-led research, we recognise the importance and validity of Te Tiriti and restate the authority of self-determination encapsulated in the concepts of mana and tino rangatiratanga. My observation is that this sounds easy enough from a Māori perspective. Of course, it is far, far more complex and much harder, because understanding Te Tiriti means that we have to understand how to share power, which hopefully I'll get to in a wee bit. Public policy is effectively anything the government chooses to do or not to do. It is a set of activities that governments engage in for the purpose of changing the economy and society. Effectively, it is a process for creating mechanisms like legislation that aim to benefit or impact the electorate in some way. This framing is important because it acknowledges that public policy extends to almost every aspect of our life. Someone has to make a determination about which issue deserves attention and how the nature of that problem is defined. The process for making decisions about what products or actions are required, some of which are exampled up there, to address these complex and salient issues, determines the rules by which opportunities are framed, what is allowed, encouraged, discouraged, and prohibited. Important if those opportunities aren't fairly distributed or adversely affect a segment of the community. My observation working for the ministry has been that policy is far more complex in-house than it needs to be. In fact, what I've noticed is that most policy people don't actually know what policy is. They pretend it's a complex specialist tool when actually it's not very complicated at all. And in fact, the complication of it, in my mind, is in some ways done on purpose to exclude. And so in my view, sometimes policy has been weaponised to exclude. Public policy is often underpinned by public opinion, as you all know, and as Anne-Marie spoke to. It's underpinned by political ideology, electoral contracts, and sometimes evidence. It is influenced by many actors, including politicians, policymakers, scientists, the public, the media. It is often reflection of the dominant society, the dominant social and ideological values and priorities. It is created to match norms, customs, and morals of the dominant culture. It uses their values, their knowledge, and their evidence. The policy space just described around the dominant leaves little room for Māori participation or Māori leadership. This is despite the fact that the Crown's mandate for creating public policy comes from Te Tiriti, in particular Article 1, which gives the Crown the right to govern. Again, my observation around policy is that the influences of policy are often politicised organisations, as many of us will know. Sometimes they work for us, sometimes they work against us. They are often embedded in institutions where the isms are a problem, racism and sexism, for example. They operate in a wider system, again, as I said, where knowledge and systems and spaces and people have been weaponised, sometimes positively, more often negatively, to create policies that are not necessarily based on the evidence, but are based on, again, the values, the norms, the priorities of dominant sectors. So within that wider space, policy space, the government has now decided to commit to working with Māori, which is fantastic, to protect taonga species, taonga works, mātauranga Māori, and in ways that support Māori rights and interests and enable Māori to derive collective benefit. The commitment aligns with government obligations under the CBD and is increasingly seen in policy instruments such as the Public Service Act 2020, the Cabinet Circular on Te Tiriti o Waitangi, and the Te Arawhiti Māori-Crown Relations numerous guidelines, all of which highlight the government's increasing comfort with and desire to be a better treaty partner, ensuring that they engage appropriately with Māori and reflect the Te Ao Māori approach in policy and decision making. Importantly, and I'll acknowledge Dame Juliet who's here, importantly you will see this kind of language on the DPMC website. So on the Department of Prime Minister's website there is numerous documents, the Cabinet Circular and the like, talking about public sector's commitment to treaty and to embedding it in policy processes and decision making. There's growing acknowledgement that drawing on a singular Western knowledge system to address complex challenges like climate change is foolish, and that Māori have a long legacy as scientists, policymakers, using our tikanga and our mātauranga to scrutinise and understand the natural world and realise collective goals. So, within that science policy interface, we have an important role as scientists to support policy. To ensure that the processes that feed international decision making are done with evidence. Good policy requires facts derived from scientific analysis and values derived from policy analysis. But there is a gap between science and policy, part of which is the availability of science in a timely manner that is directly applicable for policymakers to use. The other part is the capability of policymakers to access, interpret and apply science. I'm sure some of my other ministry colleagues who are here will acknowledge that the latter is actually a big gap, the ability of policymakers to access, interpret and apply science in their policy work. The mainstream view, though, of the science policy interface is still one that values objectivity, as I said, which is very different to the way Te Ao Māori want to be incorporated into the science policy nexus. And the approach is inconsistent with this kind of changing ecosystem that we're seeing in policy, where we want to be open to Te Ao Māori, but actually we're not amending the way that we interpret the science policy interface. So the science policy interface is a place, like I said, where scientific research can easily be shared with policymakers in digestible and timely manners, hopefully, so that they can use it to inform decision making. But what we need to remember is simply providing science does not guarantee better policy, especially if those who are accessing that science don't know how to translate it into policy speak. So I think what Anne-Marie talked about, understanding how you nuance your messages, is really important in the science policy interface and get those messages over. Again, as I noted, in the Te Ao Māori space in particular, the current science policy interface leaves very little room for Māori contribution. Perhaps that's not surprising given the wider political atmosphere that we're in, and given the kinds of language and kōrero that come out of the wider parliament, and I'm not talking about government, I'm talking about the wider parliament, and given some of the rhetoric that we continuously hear that is anti-Māori, it's not surprising that the Te Ao Māori policy science interface is still fraught. There are failings in the system. There are failings that don't recognise Māori as innovators or scientists and policy makers, and again, that's not surprising when we look at the discrimination that is in our RS&I sector right now, let alone the wider policy space. And that includes the underrepresentation of Māori within universities, the underrepresentation of Māori within CRIs, institutional racism and tokenistic approaches to funding, and the integration of mātauranga Māori into our research in inappropriate ways. So if we couple all of those issues together and couple it with a science system that is not fit for purpose, let alone the racist parts of the system that have been identified in the latest reports like Te Pai Kahurangi, it's not surprising that we really struggle in this space to ensure that we have Te Ao Māori adequately in our policy and science interface. So recently there was a report released called Te Pūtahitanga, and in that report it states that the benefits and value of Māori science within a policy making system comes from the manaakitanga that is Māori policy making. And they define manaakitanga as Māori caring for all communities, so caring for Māori communities but also for everyone else, and focus on the fact that that reciprocal relationship comes from the kaupapa Māori standpoint. And Linda Smith notes that manaakitanga alongside rangatiratanga are central ethics of a science system that works for people, not just for Māori. We know, unfortunately, the converse is not true when we think about Western or colonial policies caring for Māori communities. So we have a Māori ethic that cares for everyone, and yet we have a wider general policy ethic that cares for certain parts of the community. So despite knowing that public policy is important because it extends to almost every aspect of our lives, and determining the rules by which we operate under, despite knowing that the evidence-led policy making system that creates these instruments and actions is not working for segments of the community, i.e. the other treaty partner, and knowing that there's this growing expectation from government, especially from politicians, that we will see a Te Ao Māori view and Te Tiriti represented in these same policy instruments that aren't working, it is frustrating and interesting as well, from a science perspective, to understand why the nexus between science, policy, Te Tiriti and Te Ao Māori is not working. And how do we move towards a Tiriti-led science policy approach, which is what I was tasked with talking about. So getting to a Tiriti-led science policy space means that the Crown needs to uphold its constitutional responsibilities under Te Tiriti, and in the context of the science policy interface specifically. The growing depth of treaty jurisprudence that can help inform science policy nexus is quite complex, actually. There's the newer and wide-ranging instruments and signals I mentioned around the Public Service Act that can also help us move towards a treaty-led science policy approach. But these commitments by agencies to uphold Te Tiriti need to be more than just symbolic gestures, and I guess that's my concern, is that at the moment they're largely just symbolic gestures. Māori have repeatedly shared our aspirations for Aotearoa New Zealand. We've repeatedly asked for agencies and the Crown to give effect to Te Tiriti. And specifically, we have asked the Crown to help and ensure that we are included at decision making. What we've asked for is true partnership. One of the sayings I talk about quite a bit is that if you don't understand the treaty, think of it as a marriage or a long-term relationship between partners who actually like each other, not dysfunctional ones. In the first instance, the treaty is an agreement between partners to live together in harmony for a shared aspiration. That aspiration could be to have children and create next generation. But what we have right now is not a true partnership. We don't have a partnership where we talk about what we want to do, and one of the examples I used to give, Amanda's probably heard before, is from my own marriage. A true partnership is not when your husband takes your credit card and goes and buys a car and then tells you he bought you a surprise when it's the car you didn't want. A true partnership is usually some kind of discussion and consensus about what we're doing as partnerships. What we have right now between the Crown and Māori is effectively an abusive relationship where one partner has all the power, all the resources, and makes all the decisions and tells the other partner to be happy with it. So what we are striving for is true partnership. If we say that we want to have Māori at the table, then we truly need to share power in the relationship at an equal level. And acknowledging and incorporating mātauranga Māori into evidence and into decision making in its own right is a good start. Article 2 of Te Tiriti guarantees that Māori should have critical decision making capacity and leadership throughout the public and private sectors. Being at the table as a guest of the Crown is not tino rangatiratanga, which was promised to us in Te Tiriti. It does not lift out resources and let Māori govern them. Properly resourced science initiatives for Māori, settings that break down borders between science, policy and communities that share power, that align appropriate evidence whilst meeting community priorities and needs, and maintain and enhance Māori-centred cultural and scientific enterprise and sustainable future-focused research agendas do enhance rangatiratanga. They are Māori making decisions for themselves. So clearly it is mutually beneficial for the Crown and Māori, in my view, to find ways to work together in partnership in science policy making and to provide optimal policy settings for this to occur. This means that we need to expand what is considered legitimised knowledge and therefore what can be considered during policy development, ensuring there is a clear understanding by government policymakers of the Crown's te tiriti responsibilities to te ao Māori within the RSNI sector is important if we want to have a shared relationship. Ensuring there are greater numbers of Māori skilled and influential roles in the RSNI sector and in the science policy interface is also part of having a shared relationship. Building clearer distinction between what is Crown governed and what is Māori governed is also part of building the relational space. Working together as equals is the relational space that is talked about in Te Pūtahitanga if you want to download it and have a look. Te Pūtahitanga challenges us to move away from the kāwanatanga space, which we've always operated in, to move more further into the rangatiratanga space, but more so to move into the rangatira space, the Māori making decisions for Māori space, so that we can ultimately get to the relational space where we work as true partners, unlike my car purchasing analogy. So I think I want to kind of wrap up by saying, how do you get onto the political agenda, which is kind of what Anne-Marie was talking about as well, far more articulately than I was. A treaty-led or te tiriti-led science policy approach provides a framework that supports dual threads of Māori and the Crown working together and also autonomously. Angus McFarlane, I think he may have been involved loosely in talking to Amanda in the centre, has a really good model which leverages off the braided river. And the braided river approach is that in a braided river, we know we have tributaries that run parallel to each other or run alongside each other. Sometimes they intersect and connect. Most of the time they spend their time apart. We think about, in part, the Māori-Crown relationship like that. It's actually okay to be apart in our kāwanatanga and in our rangatira space, but it is also equally important that when we come together in that relational space, we do so as true partners, not with one holding more power. So what we're hoping for in the policy space is to ensure that we're working together in a true partnership, and that such a system would enable us to respond to key challenges facing Māori communities, facing Aotearoa and the world. Your role as researchers is to support that dual approach in the relational space and allow for power sharing with Māori. In addition, your focus should be on ensuring your research is shared in a timely manner, is accessible and in layman's terms or policy speak, addresses key needs of society or policy makers whilst not advocating for a specific issue because that will get it chucked out of the ministry, and that we move, I've already said this in part, move in a timely manner though so that we can offer actions that can be implemented now but also amended over time. And I guess what I'll probably try and wrap up on, and you can read that yourself if you want, is that one of my biggest observation working in government over the last couple of years is it's a generic observation for New Zealand as well, and I'm sure others will resonate with others. We tend to operate quite a lot in crisis mode in government. Everything is a mad panic and it must be done yesterday. It is a really bad culture, and I have been fortunate enough to do research in the disaster risk space, probably with you in part and your former colleague, and it isn't helpful to create good policy, and it isn't useful space to be in if you want to use evidence. Operating in crisis mode is natural for the government because everything is urgent and everything needed to be done yesterday. What you usually find out is my husband's an ex-builder by trade, and I used to say to people, whatever time he tells you it's going to take to build that house or to do that driveway, like take half of it and add that on again. So if he says 2 weeks, it's going to be 3. Government's a wee bit like that. We think we've only got 2 weeks to deliver something and we find out that we've got 3. But that crisis mentality means that we need, policy people want information in digestible manners that resonates with them. They can see how they can quickly pick it up and insert it into their advice or into their documents. It does mean building one-on-one relationships, and through Juliet and her team of Chief Science Advisors, you have access to ministry, so it's a really good place to share your information. But being able to work at pace is really, really important for policy people. That said, policy people like myself and my colleagues here need to learn to slow down a bit so that we can take more considered approaches to utilising evidence and better ways to create better policy that's fit for purpose. And that's probably my closing remark before I hand back to you, is that I, as a Māori in the system, as a Māori former, recovering academic, and I'm now a Māori policy maker, my frustration is that we have systems that are designed for other countries. We love to look abroad and to see what's been done overseas really, really well. But we are not Little England. We are not Little Australia. We are not, a small China. We are Aotearoa, New Zealand. We are unique because of our relationship between Māori and the Crown. What we need to be doing is research and creating policies that are fit for us. It also means that we need to know who we are and who we want to be and what we want our systems to look like so that they reflect us and our values. So, tēnā koutou katoa, ngā mihi nui ki a koutou.
Wait half a minute. Hang on. So thank you very much, Mel. Mel will answer questions on the panel, but as our guest, we are able to give you a little koha. So that was a brilliant presentation. Thank you very much. And now you can sit down.
Oh, okay, thanks. Thanks.
So it's my great honour now to introduce Professor Nick Roskridge, who is Professor in Ethnobotany in the School of Agriculture and Environment at Massey University. He's also the chairperson of the Tāhuri Whenua, the National Māori Vegetable Growers Collective. And in March, just this year, he retired after 13 years of service on the Hazardous Substances and New Organisms Committee, originally as a member of Ngā Kaihotu Tikanga Taao, the Māori Advisory Board within that committee, and which he chaired for 2 years, and then as a member of the committee since August 2014. So again, we have a wealth of academic research, applied policy analysis, and chairing of an important industry body. So kei te mihi au ki tau mana me o tau mōhio ki tēnei kaupapa, and please welcome and speak to us on your topic.
Kia ora no tātou. Now, my name is, you've just been told, is Nick. I'm originally from Taranaki. What we're going to do for the next 20 minutes is change the pace a little bit. And I know most of you are researchers, and most of you, if not all of you, want to work with Māori, right? And Māori want to work with you. But the task is that you need to understand where we're coming from as Māori, rather than research constantly knocking on the door and saying, we want you to sign off our activity because you're Māori, which is totally the wrong thing to do. So the conversation is around what are Māori looking for from the research sector, and in particular from bioprotection focused research. Okay? So we'll move away slightly from policy, because policy is what it is, but we'll have a conversation from the ground level. So what are our communities looking for, what are they striving for from the research industry? So as I say, I'm originally from Taranaki, and I'm an ethnobotanist, well actually a soil scientist by training, and an ethnobotanist by practice, and most of our work is both Māori and Pacifica, so a lot of the conversation really fits with both communities of interest. So let's have a bit of a talk. Which way do we? Oh, down there. You see, technology is faster than we are. But before we get down to tin tacks, you need from my perspective, you need to know where Māori are coming from. Ne? And it's not the same place for everybody, but there is a common factor, and that common factor is whakapapa. And it's not the task of a conference like this to try and define whakapapa, but you need to know that that is the single sole cultural factor that binds all Māori, that binds Māori to the environment, that binds the environment to Māori. So you need to understand, and if you don't know the detail of it, then you need to spend time getting familiar with the detail of it. But whakapapa is that common factor. And whakapapa really picks up on two or three things. One is it defines our origins. The second is it creates and it defines our generational obligations. And when we're talking about things like kaitakitanga, which we will do, those are obligations. They're not something that we can give or take, they're obligations. And thirdly, it supports our opportunities, because opportunities is where we want to be. We want policy, we want all of those other things to contribute and to help cement those opportunities. But from my perspective, we want to drive those opportunities as Māori. We don't want researchers telling us what the opportunities are. We want to be telling them how they can contribute to that ambition, to opportunities. Yeah. Is there one here? Yeah, I don't stand still very well. Is there one here?
We're just losing signal for the stream.
So this is a conversation. This is not set in concrete, right? But opportunity, the task, certainly from my perspective as Māori and looking at it from the outside in, is that we are looking to ensure that we are beneficiaries of the system, whatever that may look like. And opportunity is how we encase what that should be. Opportunity fits with what the government are looking for. They're trying to bring more Māori resource into some economic use. Māori want their resource to be economic as well. There are different definitions of what that economic use might be, but it's opportunity all the same. Opportunity is about contributing to the country as a whole. That's the ambition of Māori as well. We have resource. We want to make sure that we retain that resource, but it also supports us as Māori. And that's about being able to look after our whānau, look after our marae, look after our land and everything else. So the opportunity is all about that resource, but it's also about being strategic. And one of the big differences between Māori and non-Māori is that our strategic view is not just 10 years, 20 years, 30 years. It's 200 years, 300 years, 500 years. It has to be. If you look at the resource that supports us as people, then if your short-term strategy is 20 years, that's not even a lifetime. And that you need to be viewing strategic over a longer period of time. So hundreds of years, if not thousands. Everything we do is not about me, it's not about my generation, it's about those children, those mokopuna that are still to come. That's the reason we do what we do. So opportunity is about ensuring that is put into being. So to try and build your comfort zone with Māori, here's a little walk through what that looks like. Te ao Māori is both simple and complex. But in simple terms, and some of the sectors have already followed this set, there are a number of value sets, and they have been sort of positioned as cultural, physical, social, and intellectual. Nada Glavich always says, this is the physical, what you see. But when you look at me, behind me is the cultural, is the social, and is the intellectual. You don't see it, but it's there. No. So with everyone you deal with, there's a complex of culture that sits with them. And in a Māori context, there's no specific priority around what that is, but that's the complex that is Te Ao Māori. Make sense? So it's more than just dealing with the physical group. It's more than just dealing with a conversation at the table. It's dealing with all of that complex. Again, this is still in simple terms. Cultural sustainability is really about ensuring that our whakapapa is both secure, valued, and is there for the future. So that's identity. It's all of these things. It's the intergenerational respect, reciprocity. Everything that comes your way has a payback in some form. So there's always the reciprocity. It's about recognising and undertaking your responsibilities. You have responsibilities that are inherent, that are your birthright. You have responsibilities that come through your learnings and through your experience as you grow up. But it's about ensuring that that happens. It's about ensuring that you are familiar with the contemporary space and how you contribute to that contemporary space. The physical, probably the most obvious of the sustainability. From a Māori perspective, it's everything that's around you that contributes to your physical space. And that contributes ultimately to your well-being, which then contributes to the whakapapa of all those people around you. So it's all connected and it's all relative. But in modern terms, we need to deal with contemporary issues. We need science, we need policy, we need all those things to help us deal with contemporary issues. A lot of the issues didn't exist 200 years ago. The pests, the diseases, all of those things that you're familiar with are introduced issues, and we need help to be able to deal with some of those issues. So it's about context as well, and it's a responsibility for all. Social sustainability, again, the flow, that theme that flows through is about whakapapa, about ensuring that we take our whakapapa through all the elements of society, so not just within our family grouping, but wider. It's about relationships. And for all of you, as researchers, you should understand what the word relationship means, because if you want to work for Māori, with Māori, it's a relationship first. It should never be about your science kaupapa, that comes later. It's always about the relationship first. So understanding what relationships mean and how relationships come into being is important. And there is no temporal factor. So some things may take a week, some things may take a year. From a Māori perspective, it doesn't matter. It's providing the right end point or the right space becomes apparent, that's fine. So relationships don't happen overnight. Sometimes they take years to happen. But yeah, that's how it works. And if you want a long, enduring relationship with Māori, whether it's at iwi level, hapu whānau level, then you have to put some long, enduring energy into building that relationship before you start to take from that relationship. Make sense? And lastly, it's about intellectual sustainability. And this is the curved ball, I guess, in that intellectual sustainability people want to take from Aotearoa these days, from traditional knowledge. And in some ways, we as Māori are still trying to reinforce what it is we know around Aotearoa. We're still trying to regather, if you like, some of that knowledge, because it has been affected for a number of reasons, not least urbanisation and those sorts of things. So Aotearoa is not necessarily in a situation that it can be shared directly. It still has to be worked through from a Māori context. But there's constantly people try to be given out as if it's a tray of lollies. It is something to be valued. And the issue nowadays is that everything is about how you can own knowledge and how you can take advantage of that knowledge. All of the research institutes want to own outcomes. From a Māori perspective, that's not what it's about. If it's a joint process of developing those outcomes, then it's a joint process of how those outcomes move to the next phase. Lastly, I've put in here economic sustainability, because the reality of 2021 and moving forward is we are looking to participate in economic opportunity, we're looking to participate in economic stability. So, as Māori, what can we do to build that space? And you're familiar with the Manu Kahani and some of those, the Meroberry, some of those really good initiatives that are starting to flow through. They're examples of where we as Māori want to move. Not everybody's on the same page, but it's important that there is an opportunity for Māori to get the payback from the economic opportunity that contributes to our country as a whole. So recognising what economic sustainability is. From my perspective, I work in food systems. And like it or not, all of you eat every day of the year. So you all take advantage of our food systems, right? We have access to foods that we would consider Māori foods. We also partake of just mainstream foods, but all food has to originate somewhere, correct? And as Māori growers, we want to produce food that has the quality and all of the other elements about it that it can contribute to the well-being of those people around us. And if there's excess, then it goes into the market and might even go into export. But food systems are important to us. And as bioprotection experts, one of the things we're looking for is how you help us do better dealing with the new pests and diseases which weren't there in the old days. How do we participate as Māori, producing healthy foods, producing healthy produce, using inputs which have been validated potentially through both mātauranga and science, but are acceptable to our people. They're the landowners, they're the people that will work that whenua, they're the people that will invest in that whenua. So a research institution, for example, Bioprotection, we're looking for how that institute can contribute to our opportunity. It's not about that institute saying, come on Māori, we want you to tick the box because we have some work going on. It's actually about Māori saying, this is where our opportunity lies, what can you do to contribute to our opportunity? Understood that we're turning the coin, if you like, because we can, and because we should, and because that's what opportunity is about, is taking some role of control. So we grow all of these different foods. And one of the highest value components to those systems, those production that we do, is that much of it is produced in an Indigenous system. And Indigenous systems are highly marketable in that being produced in a way that is considered culturally safe and Indigenous adds value. And there's good examples of that. Tohu wines, for example. You know, wines are not a traditional food, but the system and the land and the culture values that are applied to their business ethics are a marketable factor. So it's not about running mainstream with everything. It's about taking who we are and how we do our production system and using that as a core value to the opportunity. Food systems are not always cultivated. Some of them are wild harvest. You know this food on the left, for example, is the koka. That's the cabbage tree. And even in the early settlement days with the Pākehā, they would eat that as a vegetable. But for us, it's a key vegetable. You know what this is on the right? That's karingo. That's a seaweed. How many of you eat sushi? That's the same species of seaweed that's wrapped around your sushi. But this is New Zealand seaweed. This is karingo. Traditional food, we're very familiar with it. We know when to harvest it. We know how to dry it, to cure it, all of those sorts of things. So not all of our foods are cultivated foods. Many of them are wild harvest, and some of them from the land, from the whenua, some of them from the sea. So it's trying to break that definition around what food production or what food opportunity looks like. And we won't go through this, but functional foods is one of the key sort of sellable terms at the moment. How does Indigenous knowledge contribute to functional food, to that opportunity around foods? We know, for example, if you go back to what you've just seen, the koka, the tea tree, that it won't be long and there'll be some Pākehā researchers that want to take advantage of that food type because they know it's edible, because they know it has a history of being used on the table. And the problem, certainly from a Māori perspective, is that we're not resourced to move that through into becoming a business opportunity, and often the Pākehā businesses who are resourced, who can invest in R&D, who can invest in certain things, they take advantage. The Māori potato. There's no shortage of us who grow it in small blocks, marae, home, school. One or two big Pākehā growers can usurp that from a market point of view, and you've lost the opportunity as Māori. So one of our skills is to retain opportunity at that Māori space. So the ownership of the opportunity has to sit. Okay, yep. We are just about there. So the process of that, understanding Māori horticulture is not just horticulture, but it's horticulture with values, that it has a range of crops, the traditional crops, the kumara, the hue, and understanding that the future value around that opportunity is how we retain and add value from a cultural perspective to those crops. So what is it that research can contribute into our ambition, into our opportunity? And how do we make that work for that strategic long view? The last couple of slides to remind you that we are kaitiaki. You've all heard the word, and I know you have because it's bounced around everywhere these days, but to remind you that we are kaitiaki. That it sits with us, it's an inherited responsibility. There is no one system about how it's applied. It's very unique to both place, to the time, to the issue, but to recognise that that responsibility continues. And I'm going to give you the tenets of kaitiakitanga from the late Māori Queen, Te Aotearoa. The first, to take only what you need. The second, to share the rest. To respect the limits and protect the basis of the wealth, so that the origin of your resource. And lastly, to pass on to future generations at least as good as you received. It's that simple. So the task for you, and we're not going to answer it here, but the task is to challenge yourself. How does bioprotection fit with Māori opportunity? And consider it from our space, the Māori opportunity space. No? Consider what we as Māori are looking for from the science community. How can you contribute? Part of it is about building the capacity of our people. It's not just the science, it's the experience of becoming a scientist. Having people on the inside looking out is quite valuable for the future. Having people that understand what science opportunity is alongside cultural opportunity. So there is no one hat to fit the whole space, but that's the challenge for you. So that's me for the day. Mihi atu kia koutou, and enjoy the rest of the hui. Bye.
Thank you very much, Nick. It was wonderful. So we have one more speaker in this panel, but we are going to break now to take all of the richness we have received for a drink, a cup of tea, a cup of coffee, cold drink. Can I ask you to come back out of hospitality to our online guests to be seated by 3:10, so in 20 minutes, and that way we will be on schedule for the correct time for closing. So afternoon tea is outside. Please be back. Please join me in thanking once more our third speaker, Nick, for his contribution. Well, kia ora tātou. Just a reminder of the program now that we have one more speaker, and Anita Refford I'll introduce in a moment, and she will speak for 20 minutes, and then there will be a panel of the 3 people who are able to stay for that. We aim to finish at around 4 o'clock. Those of you who are travelling to dinner tonight, please be at the overflow car park on the other side of the road. This way. Before 5:15, the buses will be leaving at 5:15. Those of you who are making your own way to Larkham Vineyard, please arrive around 5:30 for pre-dinner drinks. Any questions before we continue? No? In which case I'm very honoured to introduce Associate Professor Anita Refford, who is a graduate of this university. She did her PhD in International Trade and the Environment in the Agribusiness and Economics Research Unit here at Lincoln University. Her career took her to England, Scotland and the Republic of Ireland before returning to Christchurch, where she worked at Scion here and then has come back home to the AERU at Lincoln University. Anita is a program leader in the Deep South National Science Challenge. She is a lead author for the Intergovernmental Panel on Climate Change. She was the only New Zealand lead author for the IPCC Special Report on the Land, and she is a member of the Australasian lead writers, writing that chapter for the 6th assessment. Anita's area of expertise is the economics of climate change adaptation, and she will be leading research on that topic in the Bioprotection Aotearoa when it starts on the 1st of July. So please welcome Anita to talk to us on her topic.
Kia ora tātou. Thank you very much, Paul, for that introduction. So I'm glad that you said that I am a sort of economist, and I'm very interdisciplinary researcher, so I'm certainly not immersed in the bioprotection research. But I am excited to be in the next phase of bioprotection Aotearoa, and I'll talk a little bit about that at the end of this talk. So, climate change policy and bioprotection. When I agreed to do this talk quite a while ago, I thought I'd be talking about how climate change policy is very important for bioprotection. But as I started thinking about it closer to the time and putting my slides together, I actually realised that it's more the fact that bioprotection is really essential for climate change policy. And so for the next 20 minutes or so, I'm going to talk you through why, or to try and demonstrate why that's the case and why bioprotection research is really critical for tackling the challenge of climate change. As Paul mentioned, the IPCC produced this report on climate change and land in 2019, and there were a lot of messages in there, of course, but there were a couple, well, one really simple message really, which is that we need healthy functioning ecosystems. They underpin actually everything that we do. And I've picked out a few reasons relevant to climate change here, which is we need them to help reduce our emissions, which is what I'll refer to as mitigation, what the climate change community refers to as mitigation, as reducing emissions. We also need them to help us adapt to the impacts of climate change. So that's adapting the systems that will be affected, but also sort of using systems to help other systems adapt, and that's adaptation. And we also need healthy functioning ecosystems so that we can continue to produce the food and fibre that we in New Zealand rely on for our economic well-being and to prosper into the future. And of course we also need these healthy functioning ecosystems for a range of other reasons, including cultural, social, traditional uses of the land as well. But when we think about climate change, this underpins it. The land does. So climate change policy in New Zealand, we have the Climate Change Response Act, which is, becoming increasingly known as the Zero Carbon Act, which was amended in 2019. This is our main piece of legislation. It actually, although it's quite ambitious in a sense, we've set this target of net zero carbon by 2050, which excludes the biogenic methane, which I'm sure you're familiar with this debate that's going on in New Zealand around methane. So it's set an ambitious target, and we are one of the few countries that actually has this target enshrined in law. But when we talk about policies, we still don't really have any that are going to get us to that place of net zero by 2050. So we have this act which is actually setting the targets and is a bit more like a framework than a set of policies. But it does set the direction, and it focuses mostly on the mitigation side, but it does allow for some adaptation, which I'll come to a bit later in my talk. The other thing that the Act does is it established the Climate Change Commission, He Poarangi, and they've been in the media quite recently with the release of their final advice to government. So they published some draft advice earlier in the year, and they had a lot of submissions, a lot of feedback from different groups around that. They've revised it and presented it to the government around the setting and meeting of emissions reductions targets. And there's different sectors, it's all a lot of information in there, but land use in terms of meeting our emissions reduction target is really critical in New Zealand. So if we just look at our emissions by sector from our latest inventory, which was 2019, we can see that agriculture is the biggest source. So it's almost half of our emissions come from agriculture. I'm sure we're all aware of that. And land use change and forestry accounts for absorbing 33% of that. So almost a third is absorbed by our land use change and forestry, and which is mostly that we account for anyway, forestry. The soil does store carbon, but it's so uncertain that it's actually not really included significantly in our inventories. So between those two, the emissions coming from agriculture and the sequestration by forestry, the land is actually really critical. So currently, forests are the only option that we have for removing carbon dioxide from the atmosphere in New Zealand. Other countries, and there's research going on looking at geoengineering, sort of technological ways to absorb carbon dioxide, but currently here, these are our main mechanism. And until now, they have been our main approach to meeting our emissions reduction target. So in the Climate Change Commission's most recent advice, they actually shifted a little bit that narrative from relying very heavily on forests. So previous reports by the Productivity Commission, for example, relied very heavily on forestry to meet our commitments. The Climate Change Commission, I think, in response to all the feedback and the discussion that's been going on in the community about the conversion of land to forestry and the kind of forests that we're seeing, has really shifted to what they're now talking about, strategically managing forests in the long term, rather than being the primary goal for meeting our shorter term 2050 goal. So, and they are recommending more natives as opposed to production forests. So that's kind of interesting, but it doesn't take away the fact that at the moment forests are still our main way of absorbing carbon, and with the ETS, the Emissions Trading Scheme, being our actual main mechanism to incentivise change, they are still, and forestry is part of that, they are still a really important part of our strategy for reducing emissions to 2050, which is where bioprotection becomes really important. So you know more than I do that forests are potentially vulnerable to disease and pest outbreaks, and I really am stepping out of my comfort zone when I start talking about this, but I was looking at what kind of pests and diseases might come into New Zealand under a changing climate, and so the bark beetle apparently is something that the climate might become more favourable for that kind of pest in the future under a changing climate. And then I was looking at what it has done to forests in California, and there was a headline in some news article about 6,000 miles of climate change and this decimation that this bark beetle has caused. And if we just think about if that kind of pest comes into New Zealand and creates this kind of destruction to our forests, and our forests are our main vehicle for reducing our emissions, it's actually really putting our ability to meet those targets at risk. So the Climate Change Commission does say quite strongly that we need effective integrated pest management as we start to think about climate change. And I think we also are going to need to start managing our forests for carbon sequestration and storage as well as for timber in a way that maybe we haven't thought about it so much before. So I guess that's just to demonstrate that bioprotection is really critical and at that end of reducing our emissions, absorbing the carbon that we do emit in other sectors. So now if we move to thinking about our agricultural systems, which were the big chunk of our emissions. And within agricultural systems, livestock are the ones who generate methane, and methane is a big chunk of our emissions. So within the Climate Change Act, we've got a target to reduce biogenic methane by 10% by 2030, and then by this range by 2050, which is, I'm sure you've heard it's pretty challenging potentially, certainly to go beyond 10%. But the key point I want to make around reducing methane is that when we think about animal production and greenhouse gas emissions, the key is efficiency. So we really want to have healthy animals, which equals efficient production and lower emissions. So each animal has a sort of baseline emissions, and the more product you can get from every animal, then the less emissions, the fewer emissions you have per unit of product. So that's always been the way that New Zealand has managed to produce more product without increasing our emissions too much, is through efficiency. But again, with climate change, the pests and diseases that affect livestock are likely to increase. So things like facial eczema is going to affect their productivity, and then it's going to be harder to reduce emissions from these sectors as well. So again, I don't even know if people in the Bioprotection Aotearoa look at those livestock diseases, but that clearly, that's also another area where bioprotection is really important to enable us to reduce our emissions in our key sectors in New Zealand. Now, moving back to the Climate Change Act, it also recommended or enshrined that we have to produce a risk assessment every 6 years to identify the risks that are most important for New Zealand going forward. So the first National Climate Change Risk Assessment was published last year. I was involved in that. And it identified a range of priority risks across all sectors of the economy and society. And so one of the top 10 risks was the risks to indigenous ecosystems and species from the enhanced spread, survival and establishment of invasive species due to climate change. So that's been mentioned a bit earlier today, but it clearly is a key thing that we need to consider for our natural systems into the future. And then also, it wasn't in the top 10 risks, but another priority risks was the risk to our land-based primary sector from changes in the distribution of invasive species. So again, we can see, I've talked about how we need to reduce our emissions and how bioprotection is essential, but at the same time, we're facing these increased risks from invasive species that are able to flourish in this new climate. And that's going to challenge our production as well as our ability to reduce our emissions, as I was talking about before. So what can we do? We can adapt and try to adapt anyway. So the Ministry for the Environment, again from the Climate Change Act, in response to the risk assessment, they have to develop a national adaptation plan. And they have to do that every 6 years and within 2 years of the risk assessment. So The National Adaptation Plan will be coming out 2022. Mel, I don't know if you're involved in that at all, but that will set the direction for adaptation, I suppose, in New Zealand into the future. And I'm sure that there will be reference in there that we need to prepare for and adapt to these pests and diseases, because the research is pretty clear that there are going to be changes in their distribution in New Zealand. So I guess I'm trying to make the point that our ecosystems and our productive systems will be affected by climate change, and so we need to prepare for that. But we can actually also use ecosystems to help our other systems adapt. So things like our urban areas can be supported by nature-based adaptation or ecosystem-based adaptation. So I've got some pictures up here. I mean, ecosystem-based adaptation is basically using natural kind of systems to help protect from the impacts of climate change. So they generally have a lot of co-benefits over other types of structures. If you're thinking of a solid flood protection, a concrete, hard-engineered defence compared to something that's more natural, you're going to get the co-benefits of reducing emissions, increasing biodiversity, improving water quality, and things like that. So these examples here are sort of around what's known as green infrastructure. And we've got some stormwater protection, stormwater management pictures, and then this interesting, I just put it in there because I thought it was kind of fun, house, which is in Italy, and it's got 63 apartments in there, hundreds of trees, and the idea is that it regulates the temperature, so there's less energy required for cooling and heating. It improves the air quality, reduces the noise pollution, and generally is quite a nice, interesting place to live. So we can think about different ways to incorporate nature, I suppose, to help us adapt. So things like green spaces and trees in urban areas, which can protect from heat stress as the climate warms and things like that. And also other types of ecosystem-based adaptations might be the natural flood management that I mentioned, so restoring wetlands and floodplains. And rather than thinking about coastal sea walls, we can think about those more nature-based things. Planting trees to control erosion. Again, there's a lot of co-benefits with reducing emissions, improving water quality, increasing biodiversity. So generally, if you do the economics, and when I was in the UK, we did a study on looking at different ecosystem-based adaptation case studies, looking at the cost effectiveness. They're usually really cost effective if you can value those other benefits as well. And that's something I don't think we really talk about a huge amount in New Zealand yet, but I think there's a lot of potential. But I guess to also make the point that because they're ecosystem based, again, we need the bioprotection research to inform them. How am I doing? I'm going good. So just, I guess, reflecting on research for climate change in the bioprotection space. I think it's been really interesting today to see the diversity of people involved in bioprotection Aotearoa, which is really encouraging. I think it's really important to me to keep the big picture in mind. So I've tried to illustrate that when we think about climate change, there are a lot of different aspects to it, and some of our research might be focusing on one little area. And I think it's always important to rise up, come out of our narrow little focus, rise up, and so that we can try and maximise the benefits of the research, make those connections, and avoid any potential trade-offs and unintended consequences from just focusing too narrowly. I think it's really, really critical when we address challenges like climate change that we need these transdisciplinary approaches, including different types of knowledge, and especially including Māori knowledge and experience, because we do have so much in the room and so much that we can learn. And so I think this is really critical too. I think with climate change, it's challenging sometimes because we've got these long time frames and people can become a bit paralysed by the uncertainty. And some of the research I'm involved in is trying to support decision making under uncertainty. So yes, the future is uncertain, but we do have tools and we're developing different approaches to support decision making in that kind of context. So with flexibility, and this is illegible clearly, but it's from a biodiversity conservation paper, but basically it's looking at adaptation pathways. And the idea is that you identify different options to adapt to climate change, and you know that you'll probably need to change path at some point, and you identify the triggers that would make you change path in advance, so that when it comes time, you've got everything set up so that you can move to the next pathway, rather than locking yourself into something that might turn out to be unsuitable in the future. And then I guess to make the point, and I'm really excited that in the Bioprotection Aotearoa, the next phase, that there is more social science, but I think it is really important to remember that it's the people who make the decisions to implement the research or to take note of the research, and we've heard this in different presentations today, but from the planters, the growers, the people on the ground, right through to the national level, and the iwi and the hapu, we really need to ensure that we are integrating the physical, the natural science with the social science. So we're understanding the governance mechanisms, the decision making, the behavioural processes going on. So that's really it. I just wanted to finish by saying maybe climate policy isn't exactly bioprotection policy, but it certainly is really dependent on bioprotection to allow us to meet our climate change goals. Thank you.
Thank you very much, Anita. That's opened up a great topic for conversation. I invite you to take a place at the panel, and our two other panellists, if you're able to come forward now. The rules of the game now is I have the conch. If anybody wants to ask a question, make a statement, invite a comment, just give me a signal, and we're requested to introduce ourselves for the online audience, just saying something brief about who we are, and we'll see what this has.
Are we on? Yes, great. Thank you very much. Charles Murfield, better known as Murph from the Future Farming Centre. One of the things that's interested me today is the concept of values has been bounced around quite a bit. And a few months back now, MPI had a big hooey up in Wellington for regenerative agriculture, and they put a bunch of stuff up for us to workshop, and the room didn't like it. So we had an open mic discussion, and the conclusion of the open mic discussion, the most important thing was values. So to me, it's really interesting that this issue of value seems to keep permeating into agriculture and the discussions about how we do agriculture. And my understanding, certainly from a Maori worldview, values are right at the centre of everything. So to me, there seems to be some kind of convergence possibly even going on between a Maori worldview where values are central and the European view where the values are starting to become more and more important. So if you'd like to make some comments around that observation, that would be great.
Audience, hello online audience. Yeah, good observation, and it's in my mind not surprising, and it's not surprising in part because, like I finished in my talk, you know, when I think about what's fit for purpose for Aotearoa, there are lots of Māori culture, lots of Māori cultural components that have been absorbed into what is New Zealand culture, so not surprising in that instance. Also, humans are not, as much as all of you men in the room think you're rational, you're not. Humans are not rational, they are emotional. That we are driven by our emotions and our values that are embedded in us, you know, partly embedded in us since birth. If we were rational, we would all know that drinking is bad for us, and many of us would know that smoking is bad for us, and we would give it up because health promotion tells us to not do that. So we are driven by value sets and by emotion, and that's why it is so prevalent in policy and in government, is driven by people's value sets. So I think we've got to come to a realisation as scientists, it's much easier for us social scientists, that actually we are driven in part by our values, and our science isn't as unbiased as we like to think it is, because we are humans. And we are driven by our value set. So for me, it's not surprising. I also think that there is a strong, a moving alignment between what is largely been Māori values, you know, our values around kaitiakitanga and manaakitanga and whanaungatanga and the like, that are being widely adopted by the rest of New Zealand as well. And I always said, and this is old research, but I've always gone back to the research that Massey University did in 2000, and they got Praxis Marketing in. So Praxis is the company that does Coca-Cola's marketing and Johnson & Johnson, and Massey University was going through one of the university's restructures that we all go through a million times. And what they were trying to do was appeal to, you know, the wider New Zealand market and rebrand themselves. And they got Praxis to define what made Kiwis Kiwis. And the Listener did a big article on it, and they did the 10 characteristics of a New Zealander, and number 1 was connection to the land. And there were some other ones in there like masculinity and sports and the like, might be arguably different now. But connection to the land was the one thing that resonated, and they wrote quite a bit on that, and they said that it actually rated quite a lot higher than all the other values. And so I think that is that kind of merger between what Māori have always valued, our whenua and our wai, and what brought lots of other people to New Zealand as well.
How well does that answer your question? That's pretty comprehensive. I'll just add one thing and say that you shouldn't be scared of the word values, none of you, because you all have a set of values. Whether they're right or wrong is not for us to say, but you all carry a set of values. From a cultural perspective, we all carry, in a similar way, the same set of values, but there's a cultural layer on those values that, like it or not, is not up for grabs, and it's not up to be redefined. It's something that we carry from a cultural perspective. But don't be scared of the word, but take the opportunity to learn what other people value and how they value it, and build that into your concepts of how you move forward, especially if you want to work with communities of interest. Understand their priorities, and often it's not in the order that you think it is. It's quite often quite different. Yep. Paiana.
Well, I guess even in the more mainstream economic kind of circles, there is a bit of a recognition that until now we've really just focused on one value, which is economics. But and it's slowly coming in that we actually need to value other things. So we've got the wellbeing budget, which is a tiny step, maybe, to incorporate other values into those systems.
Okay, kia ora. Trevor Jackson from AgResearch. I was just going to say I enjoyed Anita's presentation on bioprotection and climate change. And I think it brings up the point for us that we should try and set the bioprotection agenda within the context of climate change. And I think there are many things that are almost ready to go with that. You know, we know how microbial plant associations are really important. We're planting 1 billion trees, or that's our target. How many of those are we taking the opportunity to enhance their establishment and enhance their growth by capturing that knowledge of microbial stimulation of plant growth? And in other ones, once we have trees actually growing, we are looking at capturing carbon. And at the role of established forests in that, established and regenerating, or that natural regeneration of, as we see locally with Hinawai and Banks Peninsula, is an excellent example. But from time to time, as you showed with the bark beetle, there is the necessity of operations or anticipation of those pest problems. But I'm just saying to people, and as we move forward in the next round, let's adjust bioprotection to look at it through that lens.
Thank you. I guess I would just agree with you agreeing with me, because when I was thinking about the talk, I was I started from a point of not quite sure what the connection was, and then at the end I convinced myself that this was so essential to, so I think there is definitely an opportunity to make more of that.
John Hampton, Bioprotection Research Centre, Lincoln University. Anita, you rightly stressed the importance of bioprotection to climate change, but I think it's actually bigger than that. in terms of what I call environmental change, because our production still relies on the use of chemicals. And that whole scenario is changing by legislation, by consumer requirements, and if we suddenly, it won't be overnight, but you can see what's happening. And we need to be prepared to have the tools to be able to keep producing whatever system in the absence or very much reduced ability to use, whether that's pesticides or fertilisers. And that's where I think bioprotection, sure for climate change, but it's so very important for what I'm terming environmental change, meaning the whole picture. Thanks.
I think we all agree. One of the challenges was start to look at the world from a Māori perspective, and always it's the whole of the environment rather than one aspect. Understanding climate change requires an understanding of a whole lot of other elements, so you can't isolate one aspect of science, certainly not from a Māori perspective, and you shouldn't want to. You should want to understand the drivers or the initiates, you know, the things that trigger your understanding of climate, your understanding of season. You know, they're all relevant, they're different up and down the country, all of that sort of thing, so there's a huge driver, but it's a landscape of change rather than just climate. Yep.
Kia ora. Thank you for your presentations. I really enjoyed them at the back. I'm wondering if you've got some reflections that can help me. So we've heard, and I've heard this often, that if I can paraphrase it as good mātauranga takes time. We can't just come and say, hey, what's the mātauranga view on this, and fill in a form. And it's not just the knowledge, it's the knowledge holders, and that we need to form enough relationships that we know that the person giving us the knowledge, if it's time to give us the knowledge, has enough mana to do that and speak for the community. And then we also heard from Mel that government works at pace. And there's a huge challenge getting science evidence into policy at the speed at which we need to do that. And for someone like me that's trying very hard to respect mātauranga and find ways to include it in the evidence base, how we balance that tension between needing to build the relationships and needing to provide advice and connect policymakers to the right people at breakneck speed. Sorry, it's not an easy question, but I'd love your reflections.
Well, kia ora for that. I guess if I look at it from a Māori lens, then the first thing is that the rate of interaction that institutions and all of that want from Māori is not at the same rate that Māori want to work at. And one of the big issues is that the people who are the purveyors of experience and of knowledge and all that, they're overtaxed. They've got every man in his dog knocking on the door saying, can you give us input, can you do this, can you do that? So they're being taxed time and again, and for the best part without any form of, for the wrong word, compensation. It's time given, and they do it because they want to ensure that Māori are in the conversation, but they need time to ensure that it's understood. The biggest issue in many ways is that the translation from Te Ao Māori to the science world doesn't happen easily. You know, the concepts are not easily translated across, and the scientist wants to run off with them. Kaitiakitanga, a good example. You don't learn kaitiakitanga overnight. You learn it over the generations before you have fine-tuned their experience, and then you learn from their experience and you add your experience, etc., etc. So time is important, and recognising, you know, the people I grew up with who were the holders of experience have all gone. You know, we're very much losing that old sort of experience into a more modern translation of it. So we've got to recognise there are differences, but there are different impacts on our world as well. So what the young people have to deal with is not what our grandparents had to deal with. It's quite a different physical world. You know, pests, diseases, all those things many of them are new, but also technology. All the young people now, and most of you will agree with this, they don't want to retain it up here, they want to retain it on their phone. They don't want to do the spelling like we had to do the spelling, 100 times on the board. They just want to be able to ask the phone. I mark exams sometimes and it just frustrates me that they can't spell the real basics, and they don't want to. So what does that say about the future generations? My preference is to build the base. That's the base, to give people the confidence that they can communicate without somebody not getting it. So it's time to build that base, and it's time to allow those people who are the practitioners to decide the best way to take that knowledge out there.
Can I add on to that first before you ask me a question, Matua? And so it's a really good point, because that's obviously the space where I find myself and where I want to include really awesome mātauranga by many of the people in this room, but I know that we're moving at pace that I can't do it adequately and or appropriately. And so I think that in my mind I kind of think about it as what is the long-term goal. And the long-term goal is to have mātauranga and te tiriti at the centre of all of our policy making. And then in my mind, I'm trying to work back at the moment into what are the phases that I can do that, and what is an appropriate, what can I do realistically in the next 12 months to try and get us, a small step towards the end goal. And I, in my mind, it's probably coming down to a couple of things. One is those long-term relationships that ministries just aren't good at, and the frustration for Māori is that Māori become the institutional knowledge holders of the ministries, and so it's the same Māori turning up. And I remember Matua, Jim Doherty saying this to me, that he would turn up and he would have to tell Ministry for the Environment what they said at the last meeting because all the staff had changed. So, Māori and organisations like researchers become institutional knowledge. So we have to find ways to break that. And to ensure that the relationship holds without the people. We also have to get different people into the ministry, so people who hold those relationships themselves despite where they go, people who are trusted to bring in mātauranga and to work with it appropriately and protect it. We as ministries need to have a conversation about data sovereignty and how we protect mātauranga across agencies, and who is the kaitiaki of the data, whether there is someone outside of government that becomes the custodian of Māori data. So there's a number of projects that I think we need to do so that we can then take that big step. And the other one that you alluded to that's really important to me is we need to make sure that there are different policy analysts and the like interpreting. So my biggest frustration is that when we get Māori concepts gifted to us as ministries, we love them, we think they're amazing, we take terms like kaitiakitanga that was gifted to us in the RMA 30 years ago, 20 years ago, but we completely bastardise it, and we turn it into this kind of bastardised concept that is a merger of our Western view of what it is and what Māori actually said it was, and that's not going to wash with the next generation. So what I think I said at B3, which you heard, was, the world has changed. The next generation aren't as tolerant as what we were when we sat there and listened to the kind of Don Brash speeches 20 years ago, and I don't think that kind of stuff will wash now, and I don't see it washing with the young analysts in MFE. So we have to get smarter and more sophisticated about how we're going to work at pace, but also in appropriate manners, and I don't know what the answer is, but I think I've got an idea of some things we could do at least to start there. And the other one is, and a shout out to the Māori scientists in the room, is that we need Māori scientists coming and helping in the ministries a bit more, but we just got to find a way to do that so it's safe for them as well, and that they're remunerated adequately, because, Nick, hats up to you, because you have given, a large chunk of your life for free to the Cody community, none of it which is, being paid for, but that's the kind of double duty that Māori researchers do that's not acknowledged not acknowledged or remunerated adequately. So big mihi to you for that.
Mel, you touched a real soft spot with me when you started, from where you came from, with your kōrero. Probably not so much a question, if I may. Probably to dovetail onto what you spoke about, and in particular around policy. Policy was responsible for moving Māori off their land. We know that. So, and when that happened, mātauranga Māori, as it was taught from back in our rural areas, many of that was lost. Very few retained that knowledge, and we sit here speaking of Western science, mātauranga Māori. There are only few people that truly hold mātauranga Māori as it was. I particularly, myself in particular, I grew up in that, holding that knowledge. Then I went to town, and unfortunately, it's a distant knowledge that I have of that. And sitting amongst scientists here, I feel quite humble, actually, from knowing that the people who gave me that knowledge were scientists. They were scientists in the Māori world, because they had that, and they were the holders of mātauranga Māori. We now, as we move into BA, Bioprotection Aotearoa, we're striving to retain that knowledge so that it can travel parallel with Western science. And so I'm hopeful that we're not looking at a diluted version of what mātauranga Māori was. However, and you touched on Te Tiriti o Waitangi from really the marriage. You're absolutely right, the marriage of when we come together. 181 years of marriage. We haven't progressed too well. We haven't. And so my question would be, amongst all that, is how then do we progress to a point where we sit at the table that you spoke about, and we have an equal voice at that table? I guess it depends on how big the table is, whose table it is, and knowing full well there are certain entities that try to strive for the outcome that we desire. So how then do we get to the table that can actually affect policy? Because if you don't sit at that table with policy, you got an up road.
Gosh, everyone's asking the hard questions. Because I don't have an answer for that. Thank you, Matua, for that commentary and that question. And so I want to tautoko your commentary that that it has been a long, hard 181 years, and right from the outset, policy and legislation were used as weapons against Māori. I think there's something like 161 pieces of legislation, acts of parliament that are designed to remove something from Māori, to take away language, culture, land. I come from, as you know, a number of us come from Kahanunu ki Wairarapa, where we were literally moved off of our lands under legislation and by armed forces and put on a reservation on someone else's land that was stolen from them. So certainly feel that mamai, and we're still haven't had a redress for that. We're still living on someone else's lands. And so policy and legislation leave a sour taste in our mouth, and they are hard to, as many ex-public servants, recovering public servants in the room know, it is hard to reconcile being a Māori in that space. It is hard being a Māori in that space and having to come to grips with being a representative of the Crown who did that thing to me, to my own people. So it is really hard, but we have to be in it for the long game, and realise that it's still probably going to be another, 100 odd years before we get adequate seating at that table. And like you say, it's really important to think about who picks the table and where it is and how many seats are around it and the like. And I still think there's lots of gamesmanship going on. So even the conversation about who's at the table and how big the table is gamesmanship designed to distract and designed to get Māori fighting amongst themselves about who sits at the table while the Crown does what it wants to do and needs to do at pace. So I think there's some mature conversations that need to be held amongst Māori and some mature conversations that need to be held across Aotearoa. And, you know, on the surface of it, we talk about needing to have a mature conversation around whether we want to do gene editing for predator-free, but we don't talk about whether we need to have a mature conversation about decolonisation, or whether we need to have a mature conversation about, you know, living te tiriti and embedding it in all of our institutes. And so without having those conversations and without the next generation being fully educated about our history, our rights, the tenancy agreement that we operate under in these lands, then I don't think we're going to necessarily progress in that. So we need constitutional reform, and without constitutional reform, we're not going to get adequate seating for Māori at the table ever. So all we can do is hope to get more and more Māori in the Crown space to try and get some equity for us.
I'm not sure I've got too much to add. But, you know, my mother's people come from Parihaka. And Parihaka has, for those of you, you should all be familiar with Parihaka. And one of the best things that's happened in most recent times is that our history might get taught appropriately in schools. Parihaka had a huge impact on the iwi of Taranaki, still does. And people on the outside looking in just don't get it. They don't get just how many generations have been affected, and sometimes simple things like taking away your name because they had to give themselves Pākehā names to go to school. They had to give themselves Pākehā names to do anything in society in Taranaki. Simple things like that last across many generations. So the recovery from that history will never happen overnight. But one thing about Parihaka is the willingness to embrace a wider community, the people that surround them who are not just Māori, the dairy farmers who probably the worst land managers out there in many ways, but they're part of the community that survive around the maunga, and part of the role of the people is to ensure the maunga is kept as it should for future generations. So I don't, as Mel says, it's not something that's going to be resolved in the short term, but empowerment is knowledge, and from my point of view, there's a fair chunk of New Zealand, Aotearoa New Zealand, that still needs to hear the story, to know what the history is, to know the relationship of Māori to the land. The RMA was a huge piece of legislation, and it's had a huge impact on how Māori relate to the natural resource and to the land. But until it was enacted, very few Māori had any input to it. It came out of a very non-Māori system, and then spent has spent the last 30 years trying to work out how to make it work for Māori. Still doesn't because some of those basics haven't been interpreted, haven't been carried through. But I guess what I'm saying is everything is about trying to make sure the future works. So it's not about, we need to, ko haere whakamua, we need to look back to understand what got us to this current situation, to work out how we move forward. So we should never forget our past, but we should constantly look to how the opportunity will happen. So that's my bit. Kia ora te matu.
Ka pai. Tēnā koutou ahiuria hau no Waikato. Nō reira, ko te maunga o Taupiri e mihi atu nei ki te maunga o Taranaki. Ngā wahine toa korua hoki, tēnā korua te tepu. He kōrero tāku te tuatahi ki a koe e matu ana e pāna ki te kupu kaitiaki. He hatau whakaaro e pāna ki tērā kupu. Ko te kupu e pāna ki te iwi Māori anake, ki tō tātou katoa. Mohi ona mātou te painga hoki o te tātou katoa. Engari, nā wai tērā kupu. So I better translate that, given Mel pointed to me as a recovering central government person, spending the last 2 decades in central government. Recently just switched over into plant and food research as a group general manager Māori. And so in Te Reo Māori, I picked up earlier on in Matua Nick's kōrero, the word kaitiaki. and that often you hear the word kaitiaki thrown around. And so I wondered if this was a good opportunity to explain kia whakaputa mōhio mātou, kia mātou, to explain that term kaitiaki. Fondly, we all, in parts of our mahi, refer to ourselves as kaitiaki because we have a fondness for the whenua, for the environments we live in and the rohe that we come from. But that word kaitiaki is a very important kupu. So I just asked if you could explain, I thought I'd finish with a simple question. If you could explain that term kaitiaki and who is appropriately able to use the term kaitiaki. And so also, because the other presentations have extended to the word kaitiaki, it'd be really nice to hear, Mel, an extension in terms of enshrining that in legislation, particularly in current terms where it's have regard to. And also as we move into that wider space of climate change. So, he paku pātai, just a small pātai.
Tēnā koe, Stacey.
And I should.
Good.
Stacey Whitiora. Kia ora.
Kia ora, Stacey. Tēnā koe, Stacey. Mihi atu kia koe. And no, you're not going to know exactly what kaitaki means in the next couple of minutes, but you are going to know that it's not a one phrase fits all. And it never was and it never is. The task to understand the role of kaitiaki in a contemporary space is firstly to understand what it meant in a traditional space. So what is the word, if we break down the word first, because that's how etymologists, but Pākehā, Māori like to understand words, the verb is what's important, ne? And tiaki is the verb, and tiaki doesn't translate just to guarding something. It never did, but the RMA sort of says it does. But it means drawing from all of the elements around you to be able to support the natural world that supports you. So it's a circular process of you being a part of the natural world. The term or the prefix is kai, which just means the person that feeds that verb. Ne. But it's the verb that matters. So in the old days, nobody was an expert at everything. There were tohunga who were experts and they had their space that they were experts in. Some of them were to do with conservation matters, some of them were to do with the health and well-being of the people, some of them were to do with supporting the fighting arm of the tribe. But there were tohunga who were particularly experts in their field and they were valued for that expertise, but they weren't experts in everything. And that applies today. There's no, and there should never be an expectation that any one person is a kaitiaki across everything. People have their knowledge, they have their space, they have what has been sort of expected and handed over to them through whānau, through hapu, but they're not the kaitiaki of everything. The kaitiaki of everything is iwi Māori. So we are all charged with ensuring that our natural world is at least taken forward, as Te Aotearoa said, Te Aotearoa Kau said, to take it forward in at least as good a condition as it was found. The shame for today is that everybody looks at that word and says, that's what, that's it. That's what we're looking for, and that's the term that says what we're trying to say, but no, it's not, because nobody understands the term as Māori understand the term. So if you, I don't know what I'm specifically kaitiaki of, but I do know that I have a certain knowledge space that I've been given, with both with growing up and with mahi and all of that, that it's my prerogative how that knowledge is taken forward. And so my task is to ensure that it's not used in a way that it shouldn't be used. But everything about being Māori is about a communal drive forward, supporting your community. It's never about a personal space, and it's never about one person or one individual. It's always about decisions that work for the community at large. So I don't want to define it as such, but I do want to say that it took us a lifetime of getting to this space. So you're not going to learn it overnight, because we're still learning what it is to be kaitiaki. We're still, an example of how it frustrates me. You're all familiar with the term matariki, but you're all familiar with it from a very commercial lens. Matariki now is put out in the media and on the table from a very modern, commercial, sort of contemporary lens. It actually has nothing similar to what it meant in pre-European times. Because matariki is just one element of being able to understand the lunar system, to be able to understand the stars and everything else. So it's never read just on its own. It's read over the 12 months, and it's read in context of everything else. So that's an example of how the modern contemporary world takes something, gives it a label, and everybody says, oh, woohoo, you know, here we are. Well, not here we are, sorry. But we're on the journey, and if you are prepared to spend some time and some years sort of with people who practice puanga, where I come from, who practice matariki, then you'll start to get a sense of how it works. But that's one example, and the kaitiaki is the same. So mi atu i te rangatira. And yeah, it's don't be shy to ask questions, but be respectful in what those questions are, because people don't have to give their everything, but our task is to try and make you comfortable to ask questions of us, and if it's appropriate and how we can answer, we will. Pai ana.
And I just wanted to follow up with one thing, which is, well, probably two knowing me, which alludes to your question around the use of it in legislation, or the use of it in the RM in particular. And so I have a wee bit of a personal beef with the overuse of reo in non-Māori documents and in legislation. I think in part it comes from a conversation I had with Valence Smith from AUT, and Valence said to me, you know you've been colonised when the coloniser takes your values, reinterprets them, gives them back to you, and you start spouting them. And so for me, that's kind of what kaitiakitanga has become. It's become this interpretation in the RM that was created by the Ministry for the Environment, to be fair, and has now got so much traction that even Māori spout it as meaning something that it actually, as Matua said, doesn't actually mean that. And I remember Matua Kevin Prime saying to me that he'd never even heard of the term kaitiakitanga until it was in the RMA. And so it was a term that was created, and we're seeing more and more of that in legislation. I think the intention is good. The intention is that we want to create, like I say, fit-for-purpose policies and legislation that represent New Zealand, but we're doing so without the context of where it comes from, which means we've all the potential of effectively appropriating someone else's culture again, and misrepresenting it and doing more harm again to Māori communities. So I do think that we're in a space now where we're all mature enough to kind of be a wee bit more cautious with our language and what terms we use. I know that with the increase of Māori ministers in the House, that we're starting to have these conversations about whether we should be using te reo Māori in our legislation, especially in environmental legislation where it kind of is overpopulated. So yeah, personally I've got a wee bit of an aversion to it. Not a big fan of necessarily using it. I know our mate Waitangi said to me once, just call it Bob, like just, you know, as long as it's got a definition, who gives a crap what the name is, just, you know, call it Bob. It doesn't need a flash, you know, te mana o te taio brand or something. So again, we've just got to get back to the basics and be really clear about what is the concept that we're trying to interpret or define, and being really clear about why we're using those Māori terms.
Well, thank you, Mel. I think we can all agree that all four speakers this afternoon have given a great deal of themselves, both in the preparation and the presentation and their open answers to the questions we have asked. So I ask you to join me in thanking them for what they have contributed to this symposium. Now, just 3 things before I formally close the session. The first is those who are travelling by buses, please be at the car park on the other side of the road before 5:15 so that we can leave on good time. The second, I want to acknowledge Travis and Amanda and their colleagues for their leadership and bringing us together for this conversation. And I want to thank on our behalf Sandy, who has done all of that work behind the scenes throughout the day to look after all that we need. And finally, let me ask Mr. Edwards to close the session formally with a blessing karakia. Yes.
Tēnā tātou katoa. Kei hea koe e ko, nā ura te pātai hia ki ene, tēnā koe e tuku mai tā tērā pātai. Ngari tāku e pōhehe ana i tuku ai koe i runga te mohio, i runga te kore mohio rānei. Engari pai tō pātai. Tino pai tō pātai. Nā me pēne te whakahoki atu rā kia koe. Ka whānau mai koe ki tēnei ao, he kaitiaki ai koe. He kaitiaki koe. Kaitiaki katoa tātou. I te ao katoa, nei, ka rongau ngā kōrero nei mo te whakapapa. Nāna ke tuku mai wera kōrero wainganui a tātou, mo te whakapapa. Koe rā tēnā. Ka tiaki ai tātou ngā mea katoa. Ka hoki ai ki ngā whenua ko ngā take, kia rātou ngā kaitiaki o rātou whenua. Anei, tēnei whenua o Aotearoa, nā tātou ke. Engari, ko tū hono ai tātou ki ngai Pākehā. Ko rātou anō, he kaitiaki e tūana ki o tātou nei taha. Hoino rā tērā ki a koe e ko. So, good afternoon, everyone, and what a lovely day. Does it sound like I've been awake all this time? Wonderful. Thank you to all the speakers. I just endorse what was said here, and thank you for your kōrero as well. Wonderful. So let us close this session here today, and in so doing, within this closing karakia, that we think of all of you and your families, that you are able to travel home safe to them this evening, and look forward to seeing you again tomorrow. Again, thank you to the speakers that are here and all those who delivered this morning. Tēnā tātou katoa. Kia inoi tātou. Ei hua ngā mono kei te rangi te tihi o tau Aotea, kei te mata o te whenua tau turanga wāwai kei ngā wahi katoa rā tau kororia tanga nui. E te matua e te tamata wairua tapu me ngā anahira pona tēnei rā hau tāu ponanga i chi. E tū kolopiko nei ki mōtō aroro ka tuka ke nei tēnei tamātou reo whakamoimiti Claudia Honore me ngā whakawhetai. Whakawheta atu nei mātou e i hua moau manaakitanga i uhia mai rā koe ki rungi o mātou katoa. I roto tēnei tā mātou hui i tēnei rangi. Nō reira mai rā i tōna tīmatanga tauno mai rā ki tēnei wā katuku nei ngā whakawhetai ki a koe ihua. Tēnei rā katuku ake nei ki runga tonu tō ingo tapu tō ingo kaha. Kia whakaungia mai mātou i roto tō koutou aloha mutunga kore. Kia pai here tia ki te rangi māri e inga wā katoa. Kei roto tonu ko koe te māngai te kai āwhina tautoko mai ai anei ākanei ai. Kia ora tātou.
Thank you. Yeah, very good. Well done.